तर planning A very good afternoon to everyone. Now we will begin with our session 3. Without much she worked as HOD in MLN Medical College as well as in Government Medical College in Orai till 2021. Presently, she is working as professor in Department of Biochemistry in MLN Medical College, Prayagraj. She has more than 31 national as well as international publications. Her area of interest is free radical generation and NSAIDs in rheumatoid arthritis. I warmly welcome you.
A very good afternoon to you all. So I would like to thank the IABS 22 organizing committee for giving me a chance to speak in this conference. So I'll be speaking about the original research work related to microRNA 183 cluster in HCC and its transcriptional regulation. Hepatocellular carcinoma is one of the most common primary liver tumors, and globally there is an increasing incidence of HCC, including Asia and India. It is, according to Globocan 2020, it is the third leading cause of mortality and sixth most common cancer. And the molecular pathogenesis of HCC is not completely understood. The causes for HCC mainly are viral infections with hepatitis B and C viruses, alcohol consumption, or certain toxin like aflatoxin. So chronic insult by these factors lead to necrosis and regeneration cycles in the liver, leading to the setup of the fibrosis in the liver tissue, which ultimately lead to cirrhosis, and on the cirrhotic liver HCC develops. So during these processes, there is various molecular, genetic and epigenetic alteration gets accumulated, which lead to the activation of certain pathways like those involved in angiogenesis and proliferation and lead to HCC. In recent years, deregulation of microRNAs have been known to contribute to cancer in various aspects and including HCC. So microRNAs have been studied in relation to diagnosis and prognosis of cancer, like MIP451 in case of CML. They are also marker for remission and relapse of cancer. And microRNAs can also be used as looking for the metastasis of the cancer. Like example is MIP10B in case of breast cancer. Now our interest was in the microRNA 183 cluster. So this cluster has been consistently known to be upregulated in HCC in the literature, but not much studies have been carried out to know its role in HCC. There are 3 microRNAs in this cluster, microRNA 183, 96 and 182, and they have sequence homology. They are present on chromosome 7. Now, when we started our studies, there was not much information, but now in the literature, there are certain target genes known for these microRNA members like PDC, D4, FOX1, FOX3, and their role in promotion of cell proliferation, invasion, and migration has been proposed. So we first asked the question to explore the role and diagnostic potential of this microRNA cluster in HCC. For that, we first checked their expression across a spectrum of liver cancer cell lines, various cell lines we checked, and as compared to normal cells, the expression of microRNA 96, 182, 183 was many-fold upregulated in all these cell lines. Then we went on to check, I'm sorry for the slide, so this picture is not there. We checked the expression in the HCC tissue as compared to normal tissue. HCC tissue showed very high expression of these microRNA, and as compared to adjacent tissue also, there was increased expression of all these 3 microRNA, signifying that they might have a role in the prognosis of the disease. Then by using the mimics and inhibitors of these microRNA, we checked their role in the invasion and migration by using the Matrigel assays. Matrigel assay for invasion, and we use the inserts to see the number of cells which have migrated, and we found that okay, they have a role in migration as well as invasion. As the inhibitors treated cells, they showed decreased invasion, and mimics treated cells showed increased invasion. Also, we had seen their effect in cell proliferation too. So then we went on to predict the target genes for these microRNA cluster. We used 3 softwares, TargetScan, Pictor, and Miranda. And these genes were then shortlisted on the basis of literature which were downregulated in HCC, and then targets which have role in cell viability, migration, and invasion, they were selected. Then we used microRNA mimics and inhibitors to check the expression of the predicted targets. They should show inverse expression. So I'm not sharing this data. Then we went on to do the functional microRNA mRNA interaction using inverse correlation of their expression with the microRNAs. So these were various genes which were found to be the target of these microRNA, and they have basically role in cell cycle progression, in migration, in invasion, and also DNA repair. So these genes were the targets of these microRNA 183 cluster. Then we checked their expression in various cell lines where we had found the increased expression of microRNAs, and we found that most of these target genes, they show reduced expression in this spectrum of cell lines. So from here, we could conclude that there is an inverse correlation between microRNA 96, 182, 183 with those of certain target genes like ETS1, ETR1, and LIS1. And also that this microRNA cluster may promote invasion and migration of HCC cells via negative regulation of these genes. Then next we checked the expression of this microRNA cluster in a rat model of HCC. In rat, we used DEN to create a HCC model, and we found that there is increase in the expression of these microRNA in the liver tumor tissue of in the fibrotic state, different fibrosis states of liver from this rat model. So as the fibrosis is advancing, there is increased expression of these three microRNA as compared to the F0 and F1 stage of fibrosis that was considered as 1. So there is, as a progression of fibrosis occurs, there is increased microRNA expression. And in rat serum also we could see that there is increased expression of significantly increased expression of microRNA in the later stages of the fibrosis. So it shows its biomarker potential also. Then by using various correlation and regression analysis, we could find that these microRNA 182, I'm sorry, the titles are not visible, microRNA 182 and ALT. If we use this and by plotting ROC curves, we could see that these have the power of discriminating different grades of fibrosis. Then same microRNA analysis we carried out on the chronic hepatitis subjects. So we recruited HBV infected subjects in different categories: healthy controls and inactive carrier hepatitis, liver cirrhosis, and HBV related HCC. So in all these patients, when we checked the expression of these microRNA 182 was significantly different from normal to HCC only, and it shows significant ROC curve with high area under curve. However, no change in expression of 183 was observed. But in case of microRNA 96, the expression is different in different categories, and different ROC curves could discriminate HCC from healthy subjects, HCC from hepatitis, and also HCC from the cirrhotics. So all these ROC curves showed significant area under curve. So from here we could see that, okay, this microRNA cluster has a role in cell viability, invasion, migration via various target genes. It has a role in fibrosis progression also as seen in the rat liver. And also in HBV disease, microRNA 96 and 182 show diagnostic potential. Now further, we ask the question, like, what are the transcriptional regulatory mechanisms which are involved in the overexpression of this microRNA cluster in HCC? So for this, we used this strategy. We first did the literature mining in NCBI geodatabase. And then other database, David, was used to functionally annotate the genes. Then survivor curve analysis and TCGA database was done. And then using these transcription factor binding set databases, we further shortlisted our transcription factors. So in GEO, Gene Expression Omnibus NCP GEO, it gives us the high throughput data of micro arrays, which is there in this. We searched for HCC and the differential expression of genes in HCC and normal, and we could find a list of genes which are differentially expressed in normal tissue and liver tissue having HCC. Further, when we put all these genes into database and based on the various functional characteristics like transcription, misregulation, transcription factor, transcriptional activator, we could find 19 genes whose function are either sequence-specific DNA binding, transcriptional misregulation in cancer, transcription activator, and transcriptional regulation. Now, these 19 genes were then used for the survival curve analysis by the Cancer Genome Atlas database, and we could find that aberration of these genes which are leading to decreased survival are for this set of genes. So now we had around 11 genes which showed decreased patient survival when their aberrant expression was there. Further, we use these three databases, Jasper, Contra V3, and GTRD, to see where in the promoter these transcription factor bind. So Jasper gives us position frequency matrix of the transcription binding sites, and with this we could shortlist the 6 transcription factor which actually bind to the promoter region of a set of microRNA. Then Contra V3 also gives a good score to all these transcription factors. Then the next database was gene transcriptional regulation database. Here we are putting a gene and sample ID and promoter sequence of our microRNA of interest, and we could see that okay, where these transcription factors are binding on which sequence. So finally, we have these 6 transcription factors which have their role in carcinogenesis, metastasis, and activation of oncogenes. Then we try to check their expression in different HCC cell lines and also in rat liver tumor tissue. So in different HCC cell lines, we could see that mostly they are upregulated, all these 6. And in rat tumor tissue also, we found increased expression of these genes in HCC. So based on that, okay, these 2 transcription factors are upregulated in at least 2 of cell lines and rat tumor tissue. We further shortlisted RFX5 and RNX2 to validate their transcriptional regulation experimentally. So then this strategy was followed. We cloned the promoter region of all these microRNA and their cluster also. This cluster is independently also being expressed, and all 3 microRNA have their independent promoters too. So all these promoters were cloned in PGL3 vector, and then these were transformed into E. coli cells. Recombinant plasmids were isolated. And then we have expression vectors, pCDNA expression vectors of these 2 transcription factors which were transcripted. And then we studied the Lucifer reporter assay to validate their binding to the promoter of the microRNA. So this was the vector map and the isolated plasmids, which were then confirmed by restriction, digestion, and Sanger sequencing. So this is the basic principle of Luciferase reporter assay. So when our transcription factor will bind to the promoter of the microRNA, then this luciferase gene is also coupled to the promoter. So there will be transcription and translation of this enzyme. And we will be adding substrate for luciferase, granular luciferase, and that will give us the luminescence, which will be directly proportional to the promoter activity. So by doing various protein controls and different concentration, we could see the promoter activity of microRNA 183, 82, its cluster, and 96. 96, there was no significant change, but for all other 3, there is the dose-dependent increase in the activity. So there is increased promoter activity when it was transfected with RFX5, and similarly for RUNX2. When RUNX2 was transfected, there is increase in the promoter activity of these microRNA. Further, we thought of taking it another way also. So we transfected the cells with the expression vectors carrying the transcription factors. And then we check the, okay, the transfection is successful because there is increase expression of these transcription factor in different HCC cell lines, HEP2, HEP3B, and HUH. Then we check that what is the effect of transcription of these transcription factors on the expression of microRNA cluster. So in different HCC cells, again, we studied that okay, when we are transfecting with these transcription factors, there is increase in the fold change expression of the microRNA 183, 196, 182 also. So this is in HEP2 cell. Then in HEP3B cells also we find that there is increased microRNA expression upon transfection with the transcription factors. This is for RNX2 and this is for RFX5. Still another cell line HUH7 was used and similar effects were seen. So we could find that at least at the highest concentration of the transfected transcription factor, we could see increased expression of all this microRNA. So from here we can conclude that okay, this microRNA 183 cluster, which has 3 members, 193, 96, and 182. So these are involved in these have a role in HCC as a biomarker potential. They are involved in the prognosis, and also they can depict the progression of the fibrosis. So we identified 2 transcription factor, RNX2 and RFX5, which when bind to the promoter region of these microRNAs, they lead to their increased expression of all 3 microRNAs, which further lead to increased proliferation, invasion, migration of HCC cells and inhibits the apoptosis. So now we have a clue that okay, these 2 transcription factors can also have a role in progression of the hepatocellular carcinoma along with this microRNA cluster. So I end here with the acknowledgements of my collaborator, Professor Raike Chawla. This work was carried out by 2 of my PhD students, Rajiv Chandel and Takrima Ali. And the funding by ICMR and the DPP is also acknowledged. Thank you.
Thank you, Professor Rajiv Kaur. Anyone wants to ask anything? So, thank you very much. Thank you very much, Professor Jodhi Kaur, for such an informative presentation. Our second speaker in session 3 is Professor Sadhna Sharma. I would request our judges to kindly read out this name. Professor Sadhna is Professor in Health in the Department of Biochemistry, H1 Chandigarh. He has PhD Biochemistry from the same institute. The area of research are chemotherapeutic inflammation of nanotechnology, tuberculosis and cancer, proteomic and genomic interrelationships in the diabetes and tuberculosis, drug-induced liver injury and tuberculosis, pathogenesis of osteoarthritis. She has more than 975 publications. So we welcome you, please.
So very good afternoon to all of you. So my sincere thanks to the organizing committee of IABS who has given me this opportunity to share my research in this wonderful platform. So I will begin with my talk with the title Diabetes, that is the role of diabetes in the activation, progression and pathogenesis of tuberculosis infection. So this is the research work which has been carried out by my few PhD students, which I will be presenting over here. So I will begin my talk with the WHO Global Tuberculosis Report of 2021, which indicates that globally there are 10 million people which have developed tuberculosis, and there are estimated 1.5 million deaths which have been reported in year 2021. So that indicates that tuberculosis is a very serious disease which needs to be tackled seriously. And there are a number of factors, risk factors, which are involved in the which contributes toward the progression of the disease, which includes HIV infection, undernutrition, smoking, alcohol consumption. And most important of them is the diabetes. So diabetes, which is a significant risk factor for tuberculosis, with a relative risk of 1.5 as per WHO report of 2020. And 37 million people, they develop diabetes, representing around 10% of the world's population as per the IABS report of 2021. In India, the prevalence of diabetes and tuberculosis together is 8.3% in females and 9.1% in males. And another important reason which contributes towards the development of tuberculosis is that the poorly controlled diabetic patients, they are 3 times more susceptible to develop tuberculosis as compared to those which have not been, they are not having diabetes. So the important contributing factor which is make a person more susceptible to tuberculosis because of diabetes is the immunocompromised state which occurs because of the diabetes. So chronic disease like diabetes, they increase the risk of progression from the latent to the active form of the tuberculosis. And this is because of the impairment in the immune functions which occur because of the high concentration of the glucose in both diabetic as well as the pre-diabetic conditions, indicating that both pre-diabetic as well as the diabetic subjects, they are more prone to activation of the latent form of the tuberculosis to the active disease. So what exactly is the reason which makes a diabetic person more susceptible to the infection, to carry the infection, that is a tuberculosis? Though there is a high it's a high risk factor, but exact molecular mechanism behind the association of diabetes with tuberculosis is poorly understood. So we all know that tuberculosis mainly occurs in most of the population in the latent form. Upon getting an appropriate signal, this latent form of the disease is converted into the active form. So there are a number of factors which are responsible for the activation of the disease from latent form to the active form. So this 30% of the human population globally, they have the tuberculosis infection, but only 10% develop active TB. That indicates that rest of the persons, they have the latent form. So we try to look for like what could be the probable mechanism which accounts for the activation of the latent form of the TB to the active under diabetic condition. So for this, we explored that whether diabetic is inducing some changes in the pathogen or it is inducing some changes in the host by which there is activation of the dormant state of the disease to the active form. So we try to explore the proteomic changes in the both host as well as in the pathogen. As well, we try to understand the crosstalk between the host and the pathogen, that is the M. tuberculosis F37RP. So the main aim of our study is to delineate the mechanism which is responsible for the activation of the latent form of the disease under diabetic conditions, and we try to explore the role of various dormancy or the resuscitation associated genes which might be associated with the activation of the latent form of the disease. So first we tried it in an ex vivo model by producing the in vitro human granuloma model. So in which the PBMCs, they were mixed with the extracellular matrix component containing collagen, and then they were infected with the MTB and they were allowed to grow till 9 days. So that infection was given at a different multiplicity of infection. And what we found that at a ratio of 1:0.2 MOI, the granuloma was observed as indicated by the aggregation of the macrophages surrounded by the lymphocytes. And at the ratio of 1:0.1 MOI, only microgranuloma was found and no granuloma-like morphology was seen at 1:0.5, indicating, so this was the ratio which we optimized for further experiments. And the findings were further confirmed both histopathologically and by microscopically by doing the different types of staining. So we did the hemodoxylin and eosin staining and AFB staining for the mycobacteria. And it was found that with increasing concentration of glucose, there was disruption in the granulomas, indicating that the high glucose concentration might be attributing towards the dissolution of the granuloma, because of which there could be activation of the disease. And in this, we took dexamethasone as a positive control, which is a well-known immunosuppressant. So overall from this, we found that hyperglycemic conditions affect the ability of the PBMCs to form the in vitro granuloma upon the infection with the NTB. And similarly, these conditions like observations were further confirmed by Oramin or Nile red staining. So this, what we found that, this slide is not visible. So what we found that the bacilli which were present in the granuloma, when they were subjected to higher concentration of the glucose, it resulted into the disruption of relieving or the release of these mycobacteria from the granuloma, which were further getting into the circulation, and ultimately it leads to the active form of the disease. So the overall conclusion from this experiment was that high glucose conditions, they help in the activation of the latent tuberculosis. So these experiments further prompted us. Slides are moving. Yeah.
Yes.
So till now we saw that the high glucose concentration, they have a role in the ex vivo conditions where the ex vivo model of the granuloma was found. And along with this, we found that there is a drastic increase or the drastic differential expression of the various resuscitation factors, which are ultimately responsible for the activation of the disease. So these various factors, one is the alpha-crestin, which is a well-known marker of the activation of the TB from the dormant to the active form. So there was, with the increasing concentration of this glucose, there was a down regulation of this enzyme, indicating that at higher concentration of glucose, there is an activation of the disease. Similar observation was found with some of the resuscitation factor genes, like we have a RPA-A, RPA-B, C, D, and E. So these resuscitation factors, they are ultimately responsible for the lysis of the peptidoglycans, which are present in the mycobacterial cell wall. So again, indicating that these resuscitation factors could have an important role in the activation of this disease. So then from the ex vivo studies, we moved on to the in vivo studies, where we developed an in vivo model of the latent tuberculosis by immunizing first animals with the BCG vaccine, followed by the infection with the M tuberculosis F37RP. So in the latently developed mice model, the diabetes by induced by giving intraperitoneal injection of the streptozotocin, and the establishment of latent TB was confirmed by the stabilization of the CFC counts, then formation of the granuloma histopathologically, and expression of various dormancy associated genes. So as indicated over here, that the CFU count, it was found to be stable, indicating that because the mycobacteria is residing in the dormant form, so there will not be a further replication of the mycobacteria, though they will be live. So indicating that the latent TB model has been surely formed in the animal models. So, and again in this latent mice tissue, what we found that there was a definite changes in the various resuscitation promoting factor. So this will support our ex vivo finding with the in vivo results. So then when the diabetes was given to these latent TB mice, it was found that there was increase in the CFU, indicating that as compared to the latent TB mice, there was increase in the body movement of a mycobacteria, indicating that high glucose condition or in the group where the both latent TB and diabetes are present, there is increase in the activation of the mycobacterial burden. This was observed in almost all the organs like lungs and the spleen. And further, these studies were confirmed histopathological also, indicating that diabetes is really an important risk factor which is responsible for activation of the latent TB to the active form. So these are again the expression of the various resuscitation factors. And again, the findings were confirmed through the enumeration of various colony forming units in different tissues like lungs and spleen. And it was also seen when the latent TB mice, they were subjected to diabetes-like condition and they were further treated with the various anti-tubercular drugs. They were responsive to the TB drugs, but response was not very significant. Maybe it could be because that a longer time in interval is required to see the effect of the anti-TB drugs. Though the matrix metalloproteinosis, they have an important role because they are a component of the extracellular matrix, but in our studies, we didn't find much change in the different types of the MMPs in the tissues, but in the case of serum, there was definitely significant changes in the MMP levels. So after establishing the fact that there, yes, diabetes play an important role in the activation of the disease, we try to find out exactly what are the changes which are occurring at the host level. So we try to dissect the proteomic as well as the genomic components at the host level as well at the pathogen level. So, and we found that like this study has been carried in the patients which are having only tuberculosis and in the patients which are having both tuberculosis as well as diabetes. And their proteomic analysis was done in the PBMCs. And we found that as compared to only TB subjects, there were definite change in the some of the proteins which are differentially expressed as compared to only TB patients. So, and these different subgroups of the diabetes, they have a different HbA1c levels. So from this study, we shortlisted various proteins which are differentially expressed in the TB diabetes control diabetes group as compared to only TB groups. And these proteins, they are falling into different categories which are participating in different metabolic pathways, like they are the structural proteins, they are signaling proteins, and they are participating in various metabolic processes. So this is the interactome which shows different types of the interactions which are occurring amongst different types of the proteins which are found to be differentially regulated under the diabetes tuberculosis co-pathogenesis. So under the high glucose conditions, that is the diabetic conditions, more than 10 proteins they were found to be regulated, and mostly these proteins which were upregulated, they are involved in the lipid metabolism. And what could be the possible mechanism that we studied the various cell death mechanism which could be responsible for the co-pathogenesis of these two diseases. Like it was seen that both the apoptosis as well as the necrosis, they are highly affected by the presence of different concentration of the glucose when the cells were treated with the different levels of the glucose. Then co-localization of various LAMP proteins, they were also visualized, and they were found that these proteins which are formed in the phagolysosomal fusions, they are mostly the marker proteins for the phagolysosomes. They are also found to be differentially regulated by under the conditions when the cells they have been infected with MTB and they have been cultured under different concentration of the glucose. So then moving on to the, like we have seen that yes, the diabetes is affecting the protein expression of various proteins which are present on the macrophages, and how these proteins are ultimately involved in the other changes at the genomic level. So we try to find out like what could be the possible role of the different types of the microRNAs. So microRNAs, basically they are the small microRNAs which are 18 to 24 nucleotides in length. So they have a very significant role in the regulation of the gene expression at the post-transcriptional level. So they are involved in regulation of the number of processes which include number of the immunological processes, inflammatory pathways, and the various cell death mechanisms. So far there is no any previous information which indicates the role of the microRNAs in the co-pathogenesis of TB and diabetes. So what we found that we have compared the microRNA profile of the only TB patients and the TB patients which are having dual co-pathogenesis, that is the TB as well as the diabetes. And microarray analysis indicated that a number of microRNAs they were found to be differentially regulated, which include almost 102 I think that's I'm running more than 2 minutes, right? Try to. So different types of microRNAs they were found to be upregulated or downregulated under dual conditions, that is tuberculosis as well as diabetes. So we are trying to shortlist like what is the nature of those microRNAs and what could be the possible role of those microRNAs, and is there any sort of interaction between the proteins which have been formed to be differentially regulated and the microRNAs which are also big. So we are trying to find a interaction between the different microRNAs and the proteins which could be used as a target to explore various therapeutic interventions. So by using different softwares, we have identified a number of microRNAs. So these are a list of various microRNAs which we have found to be differentially regulated. So which are involved in the number of signaling cascades. So this is both at the, when we compare diabetes versus diabetes TB or TB versus TB diabetes. So ultimately, when we just try to combine both the changes at the host as well as the pathogen and changes which occur because of the diabetic condition, which ultimately leads to the activation of the dormant form of the disease to the active form, we found that there is a significant decrease in the MMP9, upregulation of the MMP1, and there is most importantly upregulation of the resuscitation promoting factor, that is RPFP and RPFT. So these two we are thinking can be an important target to have any therapeutic implications which could be responsible for the activation of the disease. So with this, I think I would try to conclude my lectures, and I will acknowledge my mainly students. So all this work is mainly done by my 3 PhD students, Arpana Verma, Maninder Kaur and Jyoti, and few more PhD students. And I would like to acknowledge my faculty from Department of Biochemistry, Dr. Radhotra from Department of Histopathology, who has a significant contribution in the histopathological analysis, and Department of Pulmonary Medicine, from which we are recruiting the patients. And this infection work, it was done at the ICGP, New Delhi. And finally, I would like to acknowledge the financial support which I got from the ICM. So thank you once again for your patience. Thank you. So these are the few publications. Thank you.
Thank you so much, ma'am, for yet another informative presentation. Now I would request Dr. Mohammad Arshad from Aligarh. I would request our chairpersons to kindly read out the series.
Dr. Arshad. Dr. Arshad is from the Department of Aligarh Muslim University, and his area of interest is molecular endocrinology and reproductive biology, osteoporosis, as well as cancer research. And he has got many fellowships, awards, and from the plot as well as from the India I know. And he has published more than 100 publications and various national as well as international journals. So, Dr. Arshad, please. Dr. Arshad, please. Exploring cellular and molecular mechanism of osteochondrogenic potential of the thrombin phytochemicals. Dr. Arshad, please.
So first of all, I would like to thank our managing committee for providing this opportunity to present my work over here in this conference. So first of all, I would like to define what is the osteoporosis. Osteoporosis is actually a silent disease, and it occurs in the mostly in the post-menopause hormone due to the deficiency of the estrogen hormone. And it is due to the decrease of bone mineral density, due to the disturbances in remodeling of bone. The remodeling is a continuous process in the in bone, which occur by the two different type of cells, osteoblast cells and osteoclast cells. Osteoblast cells is the bone forming cell, and osteoclast is the bone resorbing cell. So the osteoblast cells that make the bone and has the ability to produce several proteins which has affinity to bind with the calcium, and that the calcium will be bind with the bone, and the bone mineral density will be increased. And there is another cell that is the osteoclast, and osteoclast is resolving cells, and osteoclast during the process of the remodeling, the osteoclast resolve the cells, and this process going on continuously. If there is the disbalance in the osteoblast and osteoclast function, or in the remodeling, formation, and resorption, there are the decrease in the bone mineral density and bone mineral loss occur, and the bone become fragile, and there is the risk of fracture. So there are treatment modalities like HRT, NSAIDs, and bisphosphonates, and they have the different side effects like dizziness, cramping, and uterine hyperplasia. So there are the need to be developed some natural products for the treatment of, or I say therapeutic essence for this osteoporosis. So what we have done, we have selected 2 or 3 different plants and for their, and evaluated their the chemical, what kind of chemicals or what kind of phytochemicals are present in this. So first we have screening problem. So one plant, its name is the Cissus quadrangularis, and it is commonly known as the harjol. Harjol means it increases the healing capacity of the bone. So in ancient time, the people they used this Cissus quadrangularis for the treatment of bone healing and increasing the strength of bone. So we did the GCMS analysis for the phytochemical estimation of this medicinal plant. Slide change. Slide change. So this is the list of phytochemicals which present in this Cissus quadrangularis. There are so many anti-osteogenic compounds and phytochemicals, antioxidant phytochemicals, and vitamins. So what we did, we isolated the osteoblast cells, primary osteoblast cells from the bone, and we cultured them, and we selected some potent markers of osteoclastic proliferation like alkaline phosphatase, and for the calcification and for the different osteoblastic markers for the gene expression. So first we did the cell viability and alkaline phosphatase activity in the osteoblast cells, and on the basis of this cell viability, we have selected some dose which have the less than toxicity as IC50. So then we have selected the 5 doses: 10 microgram, 25 microgram, 50 microgram, 75 microgram, and 100 microgram for the further estimation. So we have evaluated the raw selective oxygen species and for the nuclear apoptosis assay in the osteoblast. So our result shows that there is increase in the ROS generation as a dose dependent, and when we quantified the DCF fluorescence, then there is a marked increase of the ROS generation in this osteoblast, primary osteoblast cell. Then the apoptotic marker we have evaluated, and this is by the DCF fluorescent assay, and there are again the increased level of the ROS and apoptosis in the osteoblast cells due to the Cissus quadrangularis increasing dose. Then we have did the mineralization assay. Mineralization assay we have performed to the by the elizarine assay. Elizarine has the ability to bind with the calcium, and this shows the calcium nodule formation or calcification in the osteoblast cells. So the elizarine assays perform that there is the formation of the nodule in the osteoblast cells. So in primary osteoblast cells, with the increasing dose of the scissus coordinates, there is increase of the osteoblast calcification in the primary osteoblast cells. But again, at the higher doses, there is a decrease in the osteoblast. So there is a suitable dose is the 10 micro, 25, 50, and 70 micro m of the scissus coordinates. Then we have performed the cell cycle analysis by flow cytometry analysis, and then what we have found that this vectorial graph showing that the proportion of cell is entering into the S phase of the cell cycle. So again, there is the increased number of cells in the S phase of the cell cycle after treatment of the different doses of scissus coordinates. Then we have selected a transcriptional factor RAMEX2 for the mRNA expression. This is the RAMEX is the one of the important transcriptional factor for the differentiation of the osteoblast cells. So this analysis of mRNA expression of RAMEX, we found the upregulation of the RAMEX at the level of 25 and 50 microgram, as it is depicted by our result of the RAMEX2 and RNA expression. So this paper we have published in the Cell Proliferation. Again, we have selected another medicinal plant, that is the Punica granatum. And Punica granatum, it is a fruit that is native of the Iran, and it is used in the Indian continent and other countries. So again, these potent markers of cell proliferation of apoptosis like ALP, calcification assay, and gene expression we have performed in our experiment. And the dose 10, 25, 50, and 100 microgram, it shows that there is the increased cell proliferation in the osteoblast cells. So we performed alkaline phosphatase. Alkaline phosphatase is considered one of the most potent marker of osteoblast cells, and it contributed as an isoform in the osteoblast secretion. So osteoblastic alkaline phosphatase, which increased as a dose dependent when we treated with the Punica granatum at concentration of 10, 25, 50, and 100 microgram. So collagen deposition assay we have used in this, and collagen was one of the important proteins which provide the framework to the bone for the deposition of calcium. So the collagen deposition, again, it is found to be upregulated or increased with the different dose of Punica granatum concentration in microgram per ml. So for the mineralization assay, we again performed the calcium content analysis through the elizarine assay, and the dosage which we have given 10, 25, 50, and 100 microgram, that we found that they increased the increased the alkaline phosphatase calcification and calcium formation in the osteoblast culture. Cell cycle analysis we have performed. We have selected one lower dose and one higher dose of Punica granatum, 25 microgram and 100 microgram. And it shows that it increases the cells in the S phase in the cell cycle. So it shows that it increasing the cell proliferation rate in the osteoblast cells. And RAMEX2, as I have told, that it is a transcriptional factor which regulates the proliferation and differentiation in the osteoblast cells. So again, we found that the increase or upregulation of the RNX2 in the osteoblast cells with the concentration of immunogenic. So this paper we have published in this journal, Osteogenetic Potential of Immunogenic for Matrix Modulation, cell cycle progression and the RNX2 expression in primary rare osteoblast cells. Then we have selected a pure compound that is genistein. And genistein is a natural compound belonging to the class isoflavones, and it has the phytoestrogen activity, and this is the structure, chemical structure of genistein. So genistein, we performed the cell viability assay and we found that the cell viability increases with the 1, 5, and 10 microgram of the treatment of this genistein. Then we had performed the alkaline phosphatase activities and calcification activity for the illustrated assay. And again, we found that treatment with the 1, 5, 10 microgram concentration of genistein increases the alkaline phosphatase activity in osteoblast cells and calcification in the osteoblast cell culture, primary osteoblast cell culture. Again, we have performed the same parameters, cell cycle analysis, and which we found that this genistein increasing the cells in the S phase of cell cycle. So again, we did this ROS generation activity, and this experiment shows that the ROS generation increases with the dose of the different doses of genistein. Here we have selected 3 potent markers of osteogenesis, RNX2, BMP2, and osteocalcium. Osteocalcium is secreted a protein. It has an affinity to bind with the calcium in the bone, and it increases the calcification in the bone. So the expression of this, and we have taken a control E2 estrogen as a control. So we found that the RNX2, BMP2, and osteocalcium expression is upregulating in the primary osteoblast cells with the increased dose of genistein. Again, we have performed a function of apoptosis in osteoclast cells, and osteoclast cells, as I have told, that it is the bone resorptive cells. So there is the 2 marker which we have selected, that is the tartrate resistant acid phosphatase, one of the potent marker of the osteoclastogenesis and the apoptosis of the cell. So the treatment with this genistein decreasing the osteoclastogenesis in osteoclast cells, primary osteoclast cells. So this paper which we have published in the BMC Complementary Medicine and Therapies. Then we have selected 2 plants for the evaluation of their activities on chondrocyte cells. So again, we have isolated chondrocyte cells from the right valve area, and so this is the GCMS analysis of this solanum xanthocarbon plant which we have selected for this study, and this is the phenolic and phenolite test which are present in this medicinal plant, and DPPH inhibition assay for their for the ROS activity or reactive oxygen activities. And we perform the cell proliferation assay to find out the IC50 of this this medicinal plant. So different dosage which we have given, and then we have selected the dose of this medicinal plant. So we have selected 50, 100, and 250 microgram for our study on the basis of the viability of the test, and we have performed the cell cycle analysis and cell cycle effects analysis, which shows that there is the again increase of the cells in the S phase of cell cycle. We have performed the in vivo study, and we have developed the osteoarthritis in rat model. So we have evaluated the alkaline phosphatase activity in the rat with the different doses, higher dose and lower dose of this compound, and we found there is the decreased level of alkaline phosphatase in rat. Then we have performed the histopathological assay or histopathology of knee, and then we found that there is the increased level of and mannequin scoring systems we have performed to find out the different dissolving places of chondrocyte. Yes. So this paper which we have published in the Solomons and in the Journal of Pharmacology, then we have performed the chondrogenic potential of pumica granatum, and we find out the different chemicals in there. So again, we have performed the alkaline phosphatase activity, and we found that there is a decrease in the alkaline phosphate activities at the treatment of this. Then we have observed that there is the plateau loss of the cartilage at the knee of the rat. And then we have performed the hematotoxin, and we've seen the same histological histological marker in the bone cartilage, and it shows that there is the decrease of the cartilage. There is the increase of the cartilage resorption due to the treatment of this pumica granatum at the different doses. So collagen content we have performed. Then we have performed the proteoglycan, active collagen 2, and MMP3 activity by the gene expression analysis, and we found that there is an increased level of collagen 2 activity, MMP3 activity in the cartilage, and there is the decrease of the COX2 activity in the cartilage of due to the treatment of pumica granatum. So this paper which we have published in Environmental Toxicology. And in conclusion, we can say that the medicinal plant, they exhibit osteogenic effect by enhancing osteoblast proliferation, matrix mineralization, and other protein expression. And natural compound genistein shows the osteogenic effect by cell cycle progression and osteoblast with osteoblast attenuation. The medicinal plant, Solanum xanthocarbum and pumica granatum, which exhibit chondrogenic efficacies in chondrocyte culture and animal model studies. Further studies are needed to validate these promising natural products for their therapeutic potential in osteoarticular diseases, and other promising medicinal plants and natural products should be evaluated for their osteochondrial properties for the development of therapeutic and for the osteoporosis and osteoarthritis. So it was the aim of our study to first develop an in vitro model for the evaluation of the different compounds which have their osteogenic and chondrogenic activity. And further, we have developed an in vivo model. So we are going to perform our experiment on the in vivo model. We have developed osteoporotic model in the rat, like rectomized rat and diabetic rat. We are going to use for the further step. Thank you very much for. These are my research scholars. Dr. Sahajada is working as an assistant professor in this University in the Department of Biotechnology. Madhu Gupta, she is assistant professor in Lucknow University. Vinita, she is the assistant professor in University it is new university, which is the Siddharth Nagar, and other research scholars, they are working as a research associate in the different issue. And I acknowledge the grants funded by the different agencies. Thank you very much.
Okay. Thank you, Dr. Arshad. So any quick question, one question only. Any question? No. So our next speaker is Dr. Moinuddin. Dr. Moinuddin is a professor from the Department of Biochemistry, Jawaharlal Nehru Medical University, India. And his research interests in protein and efficacy in immunology, particularly free radical biology in relation to disease. And he has published more than 110 papers and more than 16 PhD and students. Dr. Moinuddin is going to talk studies on the dioxin modified insulin role in type 2 diabetes. Dr. Moinuddin.
Okay. Good afternoon, everybody. Good afternoon, judges, and thanks for the introduction. I would like to thank the organizing committee of this conference for inviting all of us, and we are meeting here after 2 long years. So special thanks to Dr. Mehdi for arranging this conference at such a short notice. And as we all know that he is a very dynamic person and only he can do such things. So coming to the topic, I would be speaking for these 15 to 20 minutes on some of the work carried out by my PhD student, that is in relation to the modification of insulin by the methyl glycogen. So straight away we come to glycation, and glycation all of you must be knowing, but very quickly in diabetes we know that it is hyperglycemia that is caused, and this hyperglycemia causes multiple biological events, and as the glycation gets accelerated, so it accelerates the deterioration of the biomolecules, and it leads to the structure damage of proteins, lipids, and DNA, as well as cell membrane and endothelium. And these structure damages, they have got a definite effect on the biological system, on the function of the molecules. And the glycation products, they are believed to play an essential role in the pathogenesis and progression of various diseases, and diabetes is one of them. So how the things happen? The amino group of the protein or the phospholipid or the nucleic acid, it reacts with the carbonyl group of the sugar, and it results in the formation of a shift base which rearranges to the amidori product, the early glycation product that we call, and depending on the kind of pH, whether we have got a pH less than 7, then it leads to the formation of 1,2-dicarbonase. If we have got a pH higher than 7, it leads to the formation of 2,3-dicarbonase, and finally culminating into the advanced glycation end products. And these advanced glycation end products, they have got the role in the diseases. Methylglyoxal. The methylglyoxal is it is derived from the glycolytic intermediates like dihydroxyacetone phosphate, glyceraldehyde 3-phosphates. We can also have from thyrolene. We can also have from acetoacetate and from various other pathways. And this methylglyoxal gets detoxified predominantly by the glyoxylase system that leads to the formation of D-lactate. So this is the major scavenging pathway for this methylglyoxal, because we know that methylglyoxal is highly reactive, many 20 times more reactive than glucose. And there are other minor pathways that also leads to the scavenging of this. But what is not scavenged, it reacts with the macromolecules. And when it reacts with the macromolecules, it results in the formation of advanced glycation end products. Some basic intro about the diabetes. We know that diabetes is a very dangerous disease, and the data predicts that by 2030, we would be approaching 643 million. By 2045, we would be approaching 784 million. So, over 4 in 5, that is 81% of adults with diabetes live in low and middle income group countries. And this diabetes is predicted to cause for 6.7 million deaths in 2021, the year that has passed. That means 1 in every 5 seconds. So you can very well see the effect of this disease. Type 2 diabetes, formerly called non-insulin dependent or adult onset, results from the body's ineffective use of insulin. And we know that more than 95% of the population suffers from this type, only 5% from type 1. And is largely the result of our lifestyle, because it's a lifestyle disease. So excess body weight, lack of physical activity, they are all the causes for this type of diabetes. Until recently, it was found in adults, but now it is also occurring frequently in children. There are various major complications associated with disease. We can classify them into 2 compartments, microvascular and macrovascular. The microvascular ones, they affect from eye, kidney, there are various neuropathies. Then macrovascular, we have got is increased risk of a stroke associated with brain. Dr. Rao is sitting over here. He would appreciate it better. And then we have got high blood pressure, increasing the heart. Then we have got the extremities. The peripheral vascular disease results from this narrowing of blood vessels. So this disease, in a way, it affects the whole body. Diabetes is a disease that affects the whole body. If it goes untreated, you are going to have various aftermath of this disease. The insulin molecule, how this insulin can be ligated. Insulin, we all know, has got 2 chains, B chain, 30 amino acids, and A chain with 21 amino acids. There are inter and intra disulfide bonds. So this insulin reacts with methyl glycol to give us the glycated insulin. And we have got 2 conditions in this cartoon. The upper compartment, this shows that we have got the glucose molecule. Under the normal circumstances, the insulin binds to the receptor, and then the insulin receptor substrate binds to the and form a complex. But when there is hyperglycemia, you have got the green stars. The concentration of green stars is more glucose, hyperglycemic condition. So it affects the conformation of the insulin. And when it affects the conformation of the insulin is not able to bind to the receptor, and it inhibits the binding of the insulin receptor complex. So this changes the, so what we did, we glycated the insulin with methyl glycol, taking various concentration of the methyl glycol, and we settled for 7.5 millimolar bonding on the basis of our data. And then we characterized. This talk more refers to the biophysical and biochemical characterization, and we also have got some immunological studies, and some of the patient sera have been tried for their binding to the modified and unmodified insulin. So quickly, for the animal studies, we took the rabbits and divided into 2 groups, one having the native insulin and the other one having the glycated insulin. And so I will skip this slide because it will take more time. And coming straight away to the results, we characterize this through the various biophysical parameters, and this one is the spectroscopic UV, simply UV spectroscopy. wherein we observed a hyperglycemic effect. A substantial hyperglycemic effect was observed in the case of insulin, which was treated with 7.5 millimolar methyl glycol. When we went above 7.5 millimolar, like 10 or 12 millimolar, there was no much effect, so we settled for the 7.5 millimolar of methyl glycol. So at 7.5, you can see it is 51.8% hyperchromicity at 276 nanometer. And then we did the intrinsic tyrosine fluorescent measurement. There are 4 tyrosine residues available in insulin and no tryptophan. So in this study, the quenching of the fluorescence happens when the ligand binds to the protein. And at the 7.5 millimolar of methyl glycol, the substantial quenching was observed in this tyrosine fluorescent measurement, and the level of quenching was 70.1%. Similarly, we performed age-specific fluorescence. In this case, the excitation was done at 370 nanometer, and the absorbance was taken in the range of 400 to 500 nanometer. The lambda emission was recorded at 440 nanometer. And this showed us that increased age-specific fluorescence, when we have got more and more fluorescence at 440 nanometer, it shows that there is an increased formation of advanced glycation end products. So the increase in fluorescence was 96.4% in the case of insulin plus 7.5 millimolar of methyl glycol. We did the page native as well as SDS. And in this page, in the native page, we found that there is an enhanced movement which shows the neutralization of the positive charges on lysine and arginine residues. In SDS page, we visualize some stretching of the bands, reflecting the formation of the aggregates or cross-link. Then we perform the test for the Amadori generation or the ketoamine formation through NBT assay. And the average ketoamine content of the 3 independent assays of native and glycated insulin at 4-hour incubation time was 3.56 in the case of native insulin and 141 in the case of the insulin modified with methyl glycol. So it shows that when we modify the insulin with methyl glycol, there is very high content of the ketoamine. There is very high content of the Amadori products. FTIR is a technique for the secondary structure, for assessing the secondary structure of protein, and through the amide 1 amide peak changes, we saw that native insulin gave a peak at 1657, while in the case of methyl glycol modified insulin, it was 1633, showing more formation towards the parallel beta sheet structures. In far UV, again, we recorded the negative peak at 208 and 222, which again signifies the perturbations in the secondary structure. And it gives a clear indication that there are various changes in the secondary structure of the protein which are taking place as a result of modification with the methyl glycol. From the far UV CD curve, we did the deconvolution through the BST cell, a web server for accurate protein secondary structure prediction. And we found that in the case of native insulin, the alpha helical content was 40.7, while in the MG insulin, it was 29.4. And we observed that as a result of methyl glycol modification, the alpha helical content decreased and the beta structure increased. Similarly, there was increase in the beta trans and unordered protein structure. The tertiary structure was seen with the help of near UV CD analysis, and we observed ellipticity change at 270 nanometer. In the case of native insulin, it was 1669, while in the case of insulin modified with methyl glycol, it was 2059. We check the thermal denaturation by DSC. In the case of native insulin, which shows the ordered opening of the molecule. It was the melting temperature of TM was 62.1 degrees centigrade, while in the case of modified insulin, it was 73.8 degrees centigrade. So this means that the modification is resulting in the formation of aggregates or cross links that is inhibiting the opening of the molecule. The carbonyl content is a parameter to see the oxidative damage. And when we went for the carbonyl content estimation in the case of native insulin, when we compared the carbonyl content in the case of insulin modified with methyl glycol, we found that there was an increase of 50% in the carbonyl content in the insulin molecule upon modification with methyl glycol. So there was a lot of oxidative damage in the protein. ANS fluorescence gives us the hydrophobicity parameter, and we check the ANS fluorescence. In the case of native insulin, when compared to the modified insulin showed us an increase in the intensity of around 57%. With a redshift of 15 nanometer. So this tells us that there is aggregate formation, there is cross-link formation in the case of insulin upon the modification. THTSA again showed us the same thing. It is a confirmatory test, we can say. As compared to native insulin, the insulin modified with 7.5 millimolar methyl glycol, it showed an increase of around 65%. Then we did the thermal titration analysis or thermal titration through ICT analysis. And in this case, we find that the interaction of insulin with methyl glycol results in the form you can say more of the stable structure as compared to the native molecule. Moldy-Top mass spectrometry that tells us about the mass. In the case of native insulin, we found 5808 M by S Z value, while in the case of modified insulin, it was we got two peaks, 5793 and 5906 in the case of the MD modified insulin. Then carboxymethyl lysine is a standard age advanced glycation end product. When we compared with this standard age, in the case of native insulin in LCMS, we could not find any peak corresponding to the peak of CML. But when we compared this in the case of glycated insulin, we found the peak corresponding to the peak of CML, that is the standard age, and then we can conclude that the advanced glycation end product or CML is getting formed in the case of insulin upon the treatment with methyl glycol. Then we also confirmed our results with the SEM, wherein we find the formation of the fibrils and also through transmission electron microscopic experiments that again showed the formation of the rod-like structure or the fibrils. Then we did the studies, immunization studies in the rabbits. And when we immunize the rabbits in two groups, one having the native insulin and the other one having the modified insulin, we found that the modified insulin was highly immunogenic as compared to the native insulin. We checked it with inhibition ELISA, wherein we found that in the case of native insulin, the antibodies, they inhibited the immunogen by 32% only, while in the case of the modified, it inhibited the antibodies by nearly 61%. And we did the experiment with affinity purified IgG also. In the case of native insulin, the inhibition was say 43%, while in the case of the modified insulin, the inhibition was over 70%. This all tells us that as a result of modification, it results in the formation of new epitopes that are more immunogenic, resulting in the formation of strong binding antibodies. We checked the cross-reactivity and we found that the modified epitopes were better recognized. And then we did some studies with the samples from the normal subjects as well as from the diabetes patients, type 2 diabetes patients. And in all these direct binding ELISA experiments, we found that the serum antibodies from the diabetes patients, they better recognize the epitopes on the methylgalactose modified insulin as compared to the native insulin. And it was further confirmed by the competitive binding ELISA, competitive innovation ELISA assay, wherein we got that through with native insulin as compared to the modified insulin, the native insulin, the binding was substantially low, while with the MD modified insulin, the binding was quite high. So we can conclude that the modification of insulin by methylgalactose, it results in gross structure changes in human insulin. The glycation generates highly immunogenic epitopes on native insulin, and it induces high titer antibodies in experimental rabbits. The antibodies, the induced antibodies against methylgalactose modified insulin, they show high specificity towards the immunogen as well as cross-reactivity with other methylgalactose modified proteins and amino acids. That is, the antibodies recognize the epitopes, the modified epitopes. The preferential binding of MD modified insulin by autoantibodies present in diabetes type 2 patients point towards the possible role of MD modified insulin in initiation or progression of the disease, as well as the role of MD modified insulin in disease etiopathogenesis. The MD modified insulin may serve as a possible biomarker for disease diagnosis and evaluation of therapeutic efficiency. These are some of the publications that have come out from the lab on this glycation aspect. So in the last 4 or 5 years, these are the papers that have been published. So finally, the financial assistance from UGC, CSIR, ICMR, UPCS3, DS3, and my institution, that is Aligarh Muslim University, is thankfully acknowledged. And I thankfully acknowledge my students who have carried out this work and who also prepared the presentation for us. So thank you very much. Thank you.
Thank you so much, sir. By this, we came to an end of session 3. I would request our chairpersons, Professor Subia Kareem and Professor Shyam Prakash, to kindly give away the mementos to Professor Jodhi Ford. I would request Professor Sadhna Sharma. Professor Sadhna Sharma. I would request Dr. Mohammad Arshad. Dr. Mohammad Arshad. I would request Professor Moinuddin sir to kindly take a moment. I would request Professor Salo Churamani ma'am. Professor Churamani ma'am to kindly give the memento to Professor Subia Khalil. Thank you so much, our chairpersons and Churamani ma'am, for taking the time and giving the memento. Now we will begin with the session 4. I would hand over the mic to Professor Brijesh Rathod. Thank you so much.
Thank you, Fizel. Thank you so much. I take this opportunity on the behalf of the organizing committee of IABS 2022 to introduce the chairperson for this session, Professor Moinuddin sir. He is a Commonwealth Academic Fellow, Professor of Biochemistry at JNMC Aligarh Muslim University, and his research interest, as we have already know through his presentation, he has already organized 3 international conferences at his institute, and several PhD and MPhil students got the degree, and several are working under him right now. More than 110 research articles in reputed journal he has published, and also reviewer for several editorial boards and journals of international repute. We welcome you, sir. I will now request Professor Avinash Agarwal, the chairperson for this session. Dr. Avinash Agarwal is a professor and head of Department of Critical Care Medicine at KGMU, Lucknow. He holds several administrative positions at KGMU and contributed a lot during the COVID period and saved several lives of Lucknow and in and around Lucknow. He is member of several scientific and academic bodies and published more than 100 research papers in journal of repute with high impact factor. I welcome you, sir. Please take your seat, sir. Now I request the chairpersons for this session to kindly conduct the session.
So, good afternoon, everybody. We now move to the second session post lunch, and the first speaker is Dr. Alia Shah. She is lecturer in the Department of Biochemistry in the Kashmir Institute of Medical Sciences, Srinagar. Dr. Shah has published more than 20 24 papers in peer-reviewed national and international journals. She has also got 2 chapters to her credit, and she has been awarded Best Poster Presentation Award at the symposium held at IABS University. And she was the co-organizer of 4 conferences at her institution. I invite Dr. Alia Shah to present her work, and I would request her to please limit the talk to 15 minutes. And she would be speaking on pregnancy-induced hypertension and early perspective from Kashmir Valley. Dr. Shah, please.
Thank you, sir, for the kind introduction. At the outset, I would like to thank the organizers for giving us the opportunity to share the work at the 10th IABSCON. Now, I'll be sharing my some preliminary work that I've done on pregnancy-induced hypertension from Kashmir. So pregnancy is a fundamental aspect of human condition and is definitely a special time for the mothers as well as their family. Nonetheless, the condition is associated with certain risks to the mother and the child she bears. These risks are common in every society. However, in developed countries, these risks have been largely overcome. Now, hypertension during pregnancy is a global concern and which complicates around 5 to 10% of the pregnancies worldwide. However, the percentage of pregnant women with PIH in Kashmir is much higher, almost 20%, as has been reported by one study in 2017. In fact, the percentage is even higher when we compare it with the rest of the country. Now, how do we begin to evaluate this condition? What explains the increase in the percentage of PIH cases in Kashmir? Unfortunately, there is paucity of information about PIH in Kashmir. Nevertheless, this study was designed and is perhaps the first of its kind to evaluate the condition around 3 parameters: biological, socio-economic, psychosocial, and the early predictors. Now, the study was carried out in collaboration with the Department of Obstetrics and Gynecology SKIMS, wherein patients in controls, a case was defined as a woman in the age group of 20 to 40 years in the prenatal care diagnosed by the gynecologist as hypertensive. The gestational hypertension, which is essentially described as a de novo hypertension with systolic blood pressure of 140 and diastolic greater than 190, and which manifests for the first time after 20 weeks of gestation. The control was defined as an age and gestation period post 20 weeks, matched non-retentive pregnant women. Now, this is the brief outline of the methodology that we adopted. A total of 420 patients were taken, out of which 150 were non-pregnant group, 150 were non-retentive pregnant, and 120 were hypertensive pregnant. Those attended the OPD of the Obstetrics and Gynecology department. We collected the blood samples and we assayed, performed serum analysis for oxidative stress markers. Through a questionnaire, we evaluated the socio-economic factors and other obstetric factors, and we also at present are evaluating the cytokines for in various gestational stages. Now, this table shows the age in years of the non-pregnant, the non-retentive pregnant, and the hypertensive pregnant. And also the gestational age, we took samples basically from the 3rd trimester. So for the non-retentive and for the hypertensive pregnant group. Now this table again shows the systolic and the mean diastolic blood pressure. The mean systolic blood pressure for the hypertensive group was significantly high at 148, whereas the mean diastolic was again significantly high at 96 as compared to the non-retentive pregnant at 77.3. Now, the extent of the oxidative stress, for which the marker we all know is MDA, was evaluated, and we found for the hypertensive pregnant group that the MDA lines was significantly higher as compared to their age, gestation matched and age matched non-retentive pregnant group and the non-pregnant group. We checked the level of the antioxidant enzyme, subsequently the SOD and catalase, as we know, they form the first line of defense for antioxidants, and we found that the levels of SOD was decreased as compared to the non-retentive group and their non-pregnant group. Similar was the case with catalase. This explains that as soon as lipid peroxidation happens, the first line of defense of antioxidant enzymes, both SOD and catalase, they come into play, and thus their levels are diminished in this at the first place. So we try to check out the demographic and the obstetrics characteristics of also the women. So in our setup, we did not find, though it's an established fact that the women from the rural areas have more PIH as compared to from the urban. But in our settings, because us being a government hospital, almost all women attending our clinic were mostly from the rural areas, so we could not establish any significant link. However, again, job stream, which is an established risk factor for PIH, and we again found that almost 35% of the women were employed in our cases as compared to the controls. Most of the pregnant women with PIH in our study were from the lower middle class families as compared to the middle class family, which may be because of nutritional issues, unhygienic condition, or reduced antenatal care as compared to the controls. We also got interested and tried to dig out whether there is any effect of the type of the family one resides during pregnancy. So we just interested whether the need for caring for elders, which is usually perceived to be an uneasy environment, whether that had any link with the PIH. But we did not find any such thing in our study, because maybe we are already presenting data from only one hospital. And these results cannot be as such generalized to the whole population. We also got interested because nonetheless in our culture, the habit of staying during pregnancy with in-laws, with parents' place, is more as compared to the in-laws. And yes, but we did not find any such significant difference. And as such, all members, both from the case and the controls, reported a very strong bonding with the members of the family. History of PIH, which is an established risk factor for PIH, and it was found that almost 65% of the patient women with PIH had earlier history of pregnancy-induced hypertension. Similar was the case with previous miscarriages. Almost 24% of the women reported previous miscarriages, and which can be attributed to the distress that the women face after having any miscarriage. So we also evaluated the levels for the multiple pregnancies, which is again a risk factor for PIH in subsequent pregnancy. But however, in our case, because the number of women with twin pregnancies was low, the results were not significant here. Now, family history of hypertension is again a well-established fact, and we found that at least all members in our group reported at least one of their family members being hypertensive. Same was the case with diabetes, where almost 55% of the women reported as one of their members being diabetic. Now, where do we stand as of now with regard to pregnancy research? We had a case report in our hospital wherein a 33-year-old woman, G3A2 patient with 28 weeks of gestation, complained of headache, blurring of vision, and bilateral fetal edema. She had a raised blood pressure, and she was having urine proteinuria and had almost come into the ambit of preeclampsia. Her 24-hour urines were raised, and all protein-creatinine ratio was also deranged. So on our first checkup, when visible activity was performed and hand movements in face were conducted, the optical coherence tomography was performed, which substantially showed exudative retinal detachment. Now, these were the OCT pictures wherein retinal detachment was seen. Now, this retinal detachment can also be used as a very early indicator of something getting very serious. But when I talked with my gynecologist friend and she discussed this case study, what they did was that the only modus operandi they had at that time was to stabilize the patient, monitor BP, and at the most put her on some antihypertensive drug and try to go for early cesarean section. So that's what they did, and ultimately a live baby was born. But fortunately, this patient was fortunate enough, and all these retinal detachment movements, retinal detachment processes, they got reverted back, and her vision was completely restored. However, all patients cannot be lucky as she was, and we need to develop markers which can give us early indication about the severity of PIH. So at present, we are working on cytokines and trying to establish them as some markers which can give us an indication about how bad the complication may have happened in the later part of the pregnancy. Now, this is the immune clock of pregnancy, wherein we know that cytokines, which are proteins which are secreted right from the conception to and is involved in the maintenance of pregnancy, and they contribute towards the parturition period also. What we found here is that the pro-inflammatory cytokines are increased in the first trimester, then the second trimester is involved in the enhancement of the anti-inflammatory, and subsequently in the third trimester, again the pro-inflammatory cytokines increase. So as of now, we're trying to establish immunological markers to be assessed in women with pregnancy-induced hypertension. We are assessing three cytokines, namely TNF-alpha, IL-6, and interferon gamma. Why? Because these have been usually associated with fetal demise, preterm and labor, and have also been involved in certain autism spectrum disorders and abnormal brain development in fetuses. So the strength of the study is that no such study has been reported from Kashmir, and the study will definitely help us in unraveling the nexus between cytokines and blood pressure control in pregnant women. And results will provide us an insight as to which cytokines have the potential to complicate pregnancy. Now, in general, there is a conception that pregnancy research is something that has been overlooked for decades. The reason being why it has been overlooked, the reason being in 1960s when the drug thalidomide was first of all given to women who were suffering from morning sickness. And later it was found that it had some fetal defects were developed because of the drug. So that made FDA to take some very serious note of the matter, and we enhanced regulations to the extent that almost regulators and researchers almost started excluding pregnancy-related research altogether. So there is fear of harm to the fetus, there is threat of legal liability, and complicated physiology of pregnant women. Most people try to avoid to go into such researches. And there is uncertainty about the participation of pregnant women in research and regulations which label pregnant women as being vulnerable. So almost we see most of the drugs have the warning sign, do not take this drug if you are pregnant or plan to become pregnant. So does that mean that pregnant women have to be excluded from studies? And we need to understand that lack of data can be even more harmful. So we need to change the culture of pregnancy research. But in general and in particular in Kashmir, where the burden of BIH is so high and yet the data is very, very low. Now, if we look at the expenditure spent on the pregnancy, we in our hospital have the National Health Mission, which is doing a wonderful job in terms of the Janani Suraksha Yojana, wherein incentives are provided to women from both the rural as well as the urban Kashmir. But all these funds are usually spent in terms of the antenatal care, and we need to shift the focus towards the research-oriented work. So a pregnancy research agenda needs to be developed. We need to change the mindset from excluding pregnant women towards inclusion, promote evidence-based medicine, identify questions that can be addressed through the whatever existing data is available. So a multidisciplinary approach, including that of scientists, ethicists, clinician researchers, and clinicians need to be developed. And the onus lies on the researcher. We need to change this belief that doing research on pregnant women could harm them, so it's better not to conduct the research. We need to protect pregnant patients through research and not from research. Thank you.
Thank you, Dr. Shah, for presenting a very nice lecture. The talk is open for few questions we can take quickly if there are any. So thank you very much.
The second presenter is Dr. Kamla Khan Shukla from Teams Global. Dr. Kamla Khan Shukla has done his MSc and PhD in biochemistry, and he has a professional experience. He is associate professor in biochemistry at All India Institute of Medical Science. He passed his master's in biochemistry with first division, was awarded PhD in medical biochemistry from King George Medical University in May 2020. He has worked as postdoctoral fellow at University of South Korea, in Canada, Japan, South Korea, Australia for international scientific interaction and conferences. He has published more than 40 research papers in different international journals in the area of reproductive and cancer biology, and written a book, as well as his work has been cited more than 1,600 times with an index of 80. He has received several international and national awards. He did postdoctoral fellowship from and University of Korea. He is an associate guest visitor from Frontier of Genetics and Frontier of Endocrine. He has delivered various guest lectures in international and national conferences. He is a member of national and international regulatory bodies and several scientific societies. And his area of research interest is periodical, antioxidant, cell death mechanism, environment toxicant, ubiquitin pathway, phytochemicals and non-coding RNA in reproductive cancer biology and developing biomarkers and new modalities for treatment. This topic for today's presentation is microRNA expression promotes prostate cancer stem cell expansion by targeting gene of apoptotic pathway. Dr. Kamla Khan.
Good afternoon, one and all. First of all, I would like to thank the organizing secretary who give this opportunity to present my work in this prestigious platform. So, I am going to talk about the So, my title is the microRNA expression promote the prostate cancer expansion in cancer stem cell by targeting of the apoptotic pathway. So, prostate cancer is the second most common and sixth leading cancer cause death among men in the worldwide. However, as per the WHO report, to 30,540 cases and 16,078 deaths in 2021 has been reported. Despite advancement in the treatment regimen, the prostate cancer identified and treating this disease remains challenging. Hence, it is imperative to have a marker to disease, monitor appropriate after therapy in the case of adequate treatment response. However, the dysregulation of microRNA has been implicated in tumor development, may regulate tumorigenesis via the modulating of cancer stem cell. The earlier evidence showed that the microRNA can have been directly associated with the non-tumorigenic cancer. Next. Okay. So despite the fact 21 has been also reported upregulation in the numerous human cancer, including prostate cancer. The current studies showed that the microRNA alter the signaling cascade of the facilitate the stemless potential of the cancer stem cell. Therefore, BSS, the involvement of microRNA 10B and 21 and its target apoptotic gene with characterization of prostate cancer stem cell. So here C et al. Has been reported that microRNA 21 has been associated with the apoptotic pathway gene. Another the Sekhar et al. Has been reported the micron 21 is associated with the promoted the prostate cancer stem cell. Another similar study is the micron 10B expression aggressive sequence of the cancer carcinoma and associated with the prostate cancer stem cell. Zhang et al. Has been reported the micron 10B is a potential promising biomarker for the prostate for the cancer. So here the hypothesis of what? So dysregulated expression of microRNA may affect the stemless cancer stem cell, which can be responsible for the development of progression of prostate cancer. As we know the stem cell is the cell renewal property, and normally its normal expression of microRNA cancer stem cell is the normal expression of protein codings are targeting stem cell, and then stem cell differentiation and stem cell maturation. When it get the dysregulation due to the some mutations, genetic and epigenetics, it's activate and reduce the expression of and suppress the microRNA and overexpression of oncogene. And cancer stem cell with the extensive self-renewable capacity, aberrant and differentiation, and ultimately the increase the cancer initiation, innovations and metastasis. So aim of the study is to assess the diagnostic and prognostic importance of the micron 10B and 21 in the prostate cancer stem cell through their target expression of the apoptotic pathway in prostate cancer. So objective to evaluate the expression of micron 21, 10B and its target gene expression of apoptotic pathway, and estimate the cancer stem cell CD marker 24 and 44 for the characterization of stem cancer cell. To examine the cell death parameters such as the total ROS, apoptosis, and PSA, testosterone, cytokines in prostate cancer, and lastly compare the microRNA and target gene expression along with the stem cell in different stage grade of prostate cancer subject compared with the BPS. So this is my research methodology. So I think we will take I was taken the localized and metastasis and BPS as a control. So methodology here. So I had take the blood and tissue sample from the prostate cancer patient, localized and metastasis and BPH, and then analyze the CD markers, apoptosis, total ROS, interleukin 6, TN alpha, and testosterone, and the microRNA and mRNA expression. Results. Here the demographic data. Here BPS age, PSA level, and Gleason score. This is we can see here in the localized and metastasis is significant difference as in the PSA and Gleason score as compared with the BPH. And targeting prediction of microRNA and micro 10 and 20. Why I choose this microRNA 10 and 21? So by the target predictions, we find there has relation with the apoptotic pathway. So here we can see the micron 21 and micron 21 is significantly different as compared with the BPH, and also we compare with the localized and BPS. So also we found the similar significant study. Whereas similarly we find the micron 10B and 20 micron 10B is also significant difference in the localized and metastasis as compared with the BPS, and also see the difference between the localized and metastasis. Here cytochrome C is significant increase in the localized and metastasis and also we see the difference between the localized and metastasis, but also find the significant difference. Here BCL2 and VAX is significantly decreased in the localized and metastasis as compared with the BPH. and cash space is the increase. So pro-cash space is the decrease. SMAC is also decrease in the localized and metastasis as compared with the BPS. And TNalpha and interleukin-6 is also significantly increased in the both group as compared with the BPH. And testosterone also significantly difference compared with the BPH. Total ROS is significantly increased in the localized and metastasis. Apoptosis is also significantly different. And CD markers, CD24 and 44, CD24 is increased in both group as compared to the BPH, whereas the 24 is significantly decreased in both group as compared with the BPH. And this is the overall ratio between the CD24 and 44. So significantly find the difference. So conclusion of this study, micron 21 and 10B and apoptotic pathway gene is expression like the cytochrome C, cash space was significantly higher in the metastasis PSA in the and compared with the localized. BCL2 backs and pro-cash space at SMC were the significantly decreased. The CD24 44 is a significantly higher. PSA and testosterone level and TNalpha and interleukin-6 was increased. So here we can see here this is the complete mechanism. So our finding indicate that micron 10B and 21 is likely to target apoptotic gene with crucial role in the prostate cancer pathogenesis, and it maybe have the potential role the development of the prostate cancer, and in future it might be used for the diagnostic and prognostic as a biomarker. But further study, further mechanical study is required for this study. These are references. Thank you.
Well, great presentation. Now the session is open for questions. Thank you.
And the third and the last speaker for the session is Dr. Prasul Priya Naik. Dr. Naik is Professor, Department of Physiology at IABS Jodhpur. And Dr. Naik has 50 publications to his credit with an H index of 15. He is a member of various bodies and on the editorial board of various national as well as international journals. I welcome you, Dr. Naik, for your talk. And Dr. Naik will be speaking on menaces of aluminum exposure known to unknown.
Good afternoon, everyone. First of all, I would like to thank the organizing committee of Indian Academy of Biomedical Sciences to give me this opportunity, and bestowed me the award for 2020. This is Abida Mehdi Award. I am really thankful for such nice city, nice hospitality, nice arrangement. To start with, I would like to pay homage for this noble lady. And if we go through the website of Indian Academy of Biomedical Science, it's nicely described. And I really thankful for, I feel honored to being felicitated with this award. Thank you, Indian Academy of Biomedical Sciences. Thank you, sir. For this is maybe around 30 years, I'm working with aluminum toxicity, and I don't know whether it should be said, fortunately or unfortunately, Madam Nehri, she was prey of Alzheimer's, and aluminum is a model of Alzheimer's disease, aluminum toxicity. In fact, we name any neurological disorder, any neurological degenerative disease, neurodegenerative disease, aluminum might be having some role somewhere in the process. So I thought that being in such an era of aluminum, everywhere we find aluminum, I thought that using this platform to a little update and what I'm doing for last 30 years to share my experience about aluminum. And I am afraid of aluminum. I also make you afraid of aluminum. That's what I'm trying to do. Aluminum is omnipresent. Like in art crust, it's the 8%, and topmost metal, thirdmost element. But redox inactive. Highly reactive, hardly make it available alone. Always with something, always in complete complex form, and that helps us. No beneficial use in the life process, and that's why it is believed to be biologically inert, but not necessarily. As you can see, as I'm trying to say, it's not really biologically inert. Physico-chemical properties, it's a very unique physico-chemical property. Physics and chemistry of this metal is so nice that it is being used in every place, everywhere, almost everywhere you see some or other aluminum or metal, aluminum form will be there. And it is highly melted, highly malleable, highly ductile, and it's being recycled continuously. Still, we are gradually acquiring 100 metric tons every year of aluminum. That gives us that how much aluminum we are using. As I have said, high economic impact. So we are not to say about growing grain. All of us, we are getting older, and aluminum, it takes this opportunity once it is inside the body, though there is no role of it to be inside the body and there is no pathway to get the aluminum inside the body. Still, once it is there, mostly it is there and it is affecting the nerve cells, the liver cells, the brain cells. By the way, I hope you have used toothpaste, antiperspirant, or some crystal alum after shaving, talcum powder, or these things I don't want to mention. Tea, coffee, bread, all types of bread, all types of fruit juice with preservatives. Food cooked in aluminum vessels. You might have noticed that it is, even if it is not cooked in aluminum vessel, it is the aluminum pot where it is served. Nice summer we get in aluminum pot, and it is nice. We get lot of aluminum from there. Free flow salt and nutritional supplements. All of us, we know there are 1,001 types of supplements. Some are below 7 eggs, some are 7 to 12 eggs. some are above 12, some are for pregnant mothers, some, all these supplements, they are with preservatives and with aluminum. Antacids, aspirins, these are aluminum. I hope many of you will find that you had few of them at least today. One good factor, till now I was saying that, one good factor, assimilation of aluminum is very, very restricted. Only 0.1% of the taken aluminum is internalized, systematized. However, it depends on the routes, doses, and the accompanying substances. If you have taken the lemon tea, you have taken quite good aluminum and you have given supplied citrate to get it internalized. In serum, mostly bound to transferrin, and as you know, transferrin so important substance, physiologically so important, and that is being blocked by aluminum. Kidney is very efficient to excrete, but unfortunately, aluminum is not ready to exit our body. Also, it is excreted through bile. So this is a simplified diagram where we can get, and if you notice carefully, this brain, there is no specific route to get entry into the brain. As well, there is no specific entry to get out. Actually, aluminum 3+, as you know, our body, we don't have any transport system for trivalent cations, and aluminum still gets access to inside the body, inside the system, and once it is internalized, once it is inside the cell, they have the hotspots like nucleus, lysosomes, and they remain there for lifelong, as long the cell is there and neurons are there. As already we have discussed, how we get it, it's so many ways, everything, almost everywhere, whether it's terrestrial, whether aquatic, whether plant, whether meat, whether food, all things. And there are many routes to go in. Sometimes, vaccines, about the drugs like phosphate binders, over-the-counter drugs always there, some are there. So it's a limited exit and thankfully very limited access inside the cell. Is aluminum toxic? Yes, it is. It is toxic for brain. It is toxic for spinal cord, as we know already. Neurodegenerative, neurotoxic. Liver, hepatotoxicity is proven. Osteoporosis, question mark, already came in. Aluminum may have role in osteoporosis, bone toxicity. Lungs, in case specifically, it is going through the inhalation exposure. Skin, dermal problems are there. Reproductive organ is very coming up. It's like already male part is already known. Female is doubts already will come in. And then other endocrine organs like parathyroid, pancreas, some doubts already there. Aluminum is being deposited there, but unfortunately we don't know yet what they are doing there. Immune system, questionable. Again, neuroimmunomodulatory system is the factor, could be related to the neurotoxicity. Though the main target I kept is brain and aluminum. Normally, we initial part, blood-brain barrier is not fully developed. By that time, aluminum start entering, and by the time aging, there is damage, structural damage that increase the aluminum. And as you can see, the red part, the aluminum accumulation in brain is increasing. Neuropathologies, it's a long list of neuropathology. Either aluminum is accepted as a causative factor not alone with others, or there are some association where aluminum has been found to be present on those degenerative changes. Some rat experiments, what we did, as you can see, they are nicely moving. We have given aluminum exposure for 4, 8, and 12 weeks. The first row is without aluminum, normal. Second row is with aluminum. And we have our indigenous method of tracking this. As you can see, their movement is restricted. With aluminum, we could see their normal behavioral exploring movement is restricted. Postulated mechanism, we can include in everything, almost everything. You name it, it is there, something like that. Transcription means genetic mechanism, amyloidosis, apoptosis, calcium homeostasis, cell combination, this, everything, almost everything, every possible suggestion, almost wherever there is some, everybody is trying to find it out. Does it have any role? Yes, somewhere it is there. However, oxidative stress is common, and few stalwarts who are conducting research for their life almost on aluminium, they have suggested if you see aluminium, you think of oxidative stress there. But as I have mentioned at the beginning, it's redox inactive. How it can produce oxidative stress? Then we thought of some possibilities that maybe aluminum itself is not causing oxidative stress. It is stimulating, something like catalyst. If a little bit of oxidative stress is there, aluminum is stimulating. And we found that we have done some experiment where we have given pro-oxidant, very mild dose of pro-oxidant, and we found that aluminum is stimulating oxidative stress. Then we have tried to find out how it is doing so. We have proposed that superoxide peroxide handling capacity of cells are being changed by aluminum. These are some experiments where we could see the changes in different brain region, frontal cortex, temporal cortex, thalamus, hippocampus, cerebellum, where we have separated the glutathione dependent superoxide peroxide handling capacity and glutathione independent superoxide peroxide handling capacity, and we found that aluminum is significantly altering their handling capacity, and as a result there is oxidative stress. So if pro-oxidant could stimulate the oxidative stress, antioxidant should inhibit it. That's how normal logic goes. And we found that antioxidants, in presence of antioxidant, that superoxide peroxide handling capacity is not disturbed. So antioxidant could protect it. But that was the biochemical part. When we have gone into the behavioral part, we found that antioxidant treatment couldn't protect the animals with their behavioral toxicity. So what we could suggest that oxidative stress and the neurotoxicity, they may not be really linked with each other. There may be some other pathway. So here you can see the behavioral changes. We have seen the importance of pro-oxidant and antioxidant on the aluminum accumulation. Special stains are there. You can see the cerebellar aluminum staining. You could see the Purkinje cells, how they are nicely stained with aluminum. These are hippocampal cells. Degenerative changes you could see. Same thing we have seen in zebrafish also, that behavioral, within 96 hours we could see the behavioral changes. And here we have noticed that there is a difference between male and female zebrafish behavioral alters. That gave us the idea that could be something related to estrogen, and aluminum is a metalloestrogen. Before I continue, a little bit of search on PubMed. As you can see, 2021, the number was, the 2021 September number was 3865. It is increased to 4257 and 1595. On aluminum toxicity, same way it is increased. But in case of India, it's not that improvement. We are not much bothered about aluminum toxicity. It is there, let it be there. Relatively less. Dietary aluminum exposure is relatively less. However, I am not considering those who are taking dizine regularly. Or okay, let it be. Let it be. What is there? I had a good lunch. Why not a dizine? Something like that. And there are suggestions that we have some neuroprotective, as commonly known, curcumin and other nutritional benefiting dietary ingredients, which can be important for us. Insufficient data related to aluminum exposure, lack of awareness towards the aluminum toxicity, and am I correct here? Is there a lack of awareness? I believe so. If next time if you see an aluminum foil and if you are thinking, oh, it may be toxic, I am happy with that. Identification of potential protective agents is not done properly, and unrestricted use of aluminum in medicine, on supplement food, it's unrestricted. Clinical symptom. Early symptoms. We cannot really associate these symptoms with aluminum exposure, aluminum toxicity. It can be anyone without exposure could be. Coming to later symptoms, it's so, these symptoms are so broad, we cannot really clinically classify these, okay, this is associated with aluminum toxicity. That's the problem. I think we have. So aluminum can do anything. Premature senility, infertility, renal disorders, anemia, amyloidosis, endocrine disorder, recently dyslipidemia could be. Breast cancer, last 5-10 years we are listening about breast cancer related, and neurodegenerative disorders. Recently we found that as I was talking about that aluminum is a metalloestrogen, and it may link with estrogen related, with the suggestion that male female zebrafish, they started behaving differently on exposure to the same type of condition, everything same, but their toxicity was different. Histotoxic we have done, their cytomorphometry we have done their biochemistry, they were different even in their behavior. So we thought that metalloestrogen could be one big issue, and we thought, okay, prepubertal rats and young adult female rats, both are female, and we could see that there is dyslipidemia in young adult, whereas prepubertal, same exposure of aluminum, there was no issue, no dyslipidemia. Same observation here also for plasma total cholesterol, and it is supported by the liver cholesterol and liver triglyceride there. Serum triglyceride in male female with a 15 days aluminum exposure. Obviously, this time it was intraperitoneal. And you could see that male group, control and treated, not much different. but control and treated female group. We could find that even the peripheral nervous system, autonomic nervous system, we could see that RR interval is changing. We could see that total power for autonomic reactivity changing. So what we are suggesting here, that we might be coming towards a new way of thinking, taking the aluminium in a different directive, where estrogen mediated pathway may be a crucial factor, where we can link that neurodegeneration, we can link the osteoporosis, we can link the male-female behavioral differences, what experimentally what we are seeing. So what we can do? Identify the avoidable sources of aluminum exposure and keep them away from us. It is very difficult. Everywhere we find aluminum, and almost we have to stop everything if we think of avoiding aluminum. Thank you very much. I acknowledge my students as well as my host institute. Current host institute is IABS Jodhpur. Before that, I was working in NRI Institute of Medical Sciences and Guntur. I acknowledge them, and I also acknowledge my funders, DBT, DRDO, ICMR, and obviously host institute intramural funding I have received. Thank you very much. Thanks for your presence.
Thank you, Dr. Naik, for a nice talk on aluminum toxicity. We can have one or two quick questions if there are any.
Yes, sir. Please.
What about the interaction of aluminum with lead or any other material?
Sir, I am so afraid of aluminum alone. I couldn't try other things, but that's a good suggestion we can think of.
What you're doing for awareness?
As such, I have not done anything. I whenever I get an opportunity, I just make my audience afraid of aluminum. Nothing else I can do.
Someone
I know.
lead, and I'm sure lead is not toxic.
I do agree. I do agree.
It is entering in the body. I feel that it is negligible.
Yeah, it is negligible. But when as the medical science is growing, our life expectancy is growing. So aluminum exposure is also growing. That's the problem.
Really the body, and what kind of damaging changes it takes.
All the neuropathology, somewhere it is there. But unfortunately, we are not able to identify what it is doing. I'm trying my best. That's
But try to do something.
yeah.
You'll also do it.
Yeah. Thank you.
Thank you, Dr. Naik. So,
Thank you, sir.
To the end of this session, over to the organizers.
Thank you so much, sir. And thanks a lot to all these three speakers, Dr. Alia, Dr. Kamlakant, and Dr. Prasoon Priya, for their enlightening talk and enlightening all the audience with their scientific findings. I'm thankful on the behalf of IABS committee to the chairpersons for the smooth conduction of the session. May I now request the chairpersons to kindly do the honors to the speakers. May I request Dr. Alia, please. Again, madam. Dr. Kamlakant, please. Dr. Prasoon, please.
May I request Dr. BSS Rao, Registrar NIMHANS, Bangalore, to kindly do the honors to the chairperson, Dr. Avinash Patwal.
Thank you very much.
Thank you so much.
Thank you very much.
Now, I'll request the next session convener, Dr. Siraj and Dr. Shadab, to kindly start the session. Dr. Siraj.
It's there actually.
We're waiting for you, Shadab.
स्टार्ट हम आपको बुला लेंगे
और इधर ही माइक दे दो, the hand will sit here.
या
So we have a page of
Okay, hello everyone. Now we are going to start session number 5. That is Professor Nurul Islam oral presentations, and the participants are not much in this. Only 6 participants are there. Let me introduce their names to you. First is Dr. Priyatma Ames, New Delhi. Yeah, you keep sitting. Dr. Suman Mishra from SDPGI, Lucknow. Third is Dr. Meenakshi Sharma from AIMS, New Delhi. Fourth is Dr. Priyadarshini G, Jipmer, Puducherry. I think she will be joining us online. And Dr. Upinder Singh from AIMS, New Delhi. And Dr. Shruti CR from CSIR from Thiruvananthapuram. So, Dr. Shruti is here? Yeah, that's great. Okay, now I would request Dr. Shadab Raza. He is the head of the STEM Cell Laboratory in University, to introduce the judges.
So a very good afternoon to all of you. In this session, we have 4 esteemed examiners. And the first one is Dr. Tupil Venkatesh. Dr. Tupil Venkatesh is a professor emeritus at St. John's Medical College, Bangalore. He is the CEO and the director of the Foundation for Quality India and the director of the National Referral Center for Lead Project in India. Dr. Tupil is currently a national chairperson of the Indian Society for Lead Awareness and Research, that is in SLA. And honorarily, we call him the lead man of India. Lead man of India. Yes.
He's doing lead man
on the left. Lead man. Yes. So, and the next person is Professor Afrozul Haq. I believe he is not here. He has left. So the next 2, Shiraz will introduce the next 2 examiners. Thank you.
very brief. Yeah, sure. It's very long list of awards, and it's very long. So I'll keep it brief, but we are truly privileged to have such distinguished, stalwarts, is the right term, or the elite persons in your field. So you're welcome, sir. And the next judge is Dr. B.S. Shankaranarayan Rao. He is FINS, FABS, and he is also professor of neurophysiology. And he is the coordinator of Neurobiology Center, Research Center, and office in charge in Central Animal Research Facility, faculty in charge Electrophysiology Laboratory, National Institute of NIMHANS, Institute of National Importance in Bangalore. Sir, I think if I go on with reading the awards, just to be in short, he has received innumerable awards and honors. He is fellow of Indian Academy of Neurosciences and so many. And lastly, I would like to introduce our next judge, Dr. Sudhir Kumar Goyal, sir. He is awarded with fellowship he is professor and head Department of Biochemistry in T.S. Mishra Medical College and Hospital in Lucknow. And he has been awarded with fellowship A4U by Spanish government to visit 4 universities for different collaborative projects. He is also a CSIR Junior Research Fellow way back in 1976. Sir, should we start?
Yeah, sure.
Yeah, okay. So now I would like to request our first speaker, Dr. Priyatma from IABS New Delhi, to come and present her talk.
So the general comments and also the guidelines. So this is the 6 presentations. So the duration of the presentation is for 10 minutes. At the end of 8 minutes, there will be a bell, and you will have 2 minutes. So you will have the questions need to be answered. Okay. So keep the time, and this will be evaluated for a neural discount for test oral presentation.
A very good evening to all the respected faculty members and my dear colleagues. I am very thankful to the IABSCON Society for giving me this opportunity to present my work. My work is on dysregulation of dimethyl arginine and its association with claudin 7 and claudin 18 mRNA and PD-L1 expression in gastric cancer. It is not moving.
Yeah.
Now, why is gastric cancer? Why the work should be on gastric cancer? As we all know that gastric cancer is the 5th most commonly diagnosed cancer, and the 4th most common cancer related is due to this cancer, according to GloboCan report. And around 5.6% new cases are worldwide due to this for this GC, and deaths of 7.7% is because of this GC. And in India, only 34,000 new cases are reported every year, according to ICMR consensus report. And now, what is gastric cancer? It is a multifactorial processes are involved, which causes various genetic and epigenetic changes that leads to malignancy of the cell. And it mainly occurs in the mucosa, and 85% of the gastric cancer are mostly adenocarcinoma that occurs in the lining of the mucosa. And 40% develop in the pylorus, and 40% develop in the pylorus, and 15% is the upper cardia, and 10% metastasizes to the other parts of the organs. Various factors are involved for the gastric cancer, which leads to the superficial gastritis, which is mostly avoided by the, if a person suffers from gastritis, it mostly avoids just like a gastritis symptom. But it ultimately leads to the chronic inflammation, and then the atrophic gastritis occurs. And as a result of various factors, metaplasia occurs, dysplasia ultimately leads to the gastric cancer. Now, various research questions are there whether this ADM, asymmetric dimethyl arginine, is associated with the development of this gastric cancer. Then, what is the role of this nitric oxide in the development of gastric cancer? And is nitric oxide is related to the mitochondrial mode of action, mitochondrial pathway or apoptotic pathway for the gastric cancer development? Or we have focused on the tight junction proteins, that is 7 and 18, whether it is also having role in the gastric cancer development, or it progresses to the MMP7 dysregulation or not. And one more factor, PD-L1, what's the role of PD-L1 in gastric cancer? So we have focused on these areas, ADMA, nitric oxide, then the mitochondria, and then the PD-L1, and the tight junction proteins, and MMP7, which is the extracellular matrix protein. So we hypothesizes that either changes in the level of ADMA and expression of claudins and nitric oxide and PD-L1 and matrix metalloproteinases and mitochondrial DNA copy number alone or synergistically leading to the gastric cancer or not. So our objectives are to compare the levels of ADMA, SDMA, nitric oxide. In all the cases, we have recruited 3 cases: gastric cancer cases, disease control, and healthy controls. And we have to determine the mitochondrial DNA copy number in all the cases. Then we have checked the mRNA expression of claudin 7, 18, and PD-L1 in gastric cancer cases. This is the cross-sectional study, and on the basis of the power factor, we have calculated the number of the subjects, and inclusion and exclusion criteria we have taken for each subject. And gastric cancer cases were mostly the naive cases we have recruited. And for sample collection, we have collected the blood and biopsy of the cancer patients and disease controls. This is our work plan. We have recruited all the cases, controls, disease control, and gastric cancer cases. And for controls, we took the blood samples. For disease control, we took the blood and biopsy. And for the gastric cancer cases also, we took the blood and biopsy. And we did the assessment of various parameters. ADMA, SDMA, nitric oxide, we have done ADMA, SDMA by HPLC, nitric oxide by spectrophotometry, and mitochondrial DNA expression and nuclear DNA was done by real time. And then the claudin 7, 18 expression and PD-L1 expression was seen by real time PCR. Coming to results, our results, we have found that the all was age and sex matched, but in gastric cancer cases, 75% of cases were reported of male, and 5% which we have reported were 25% were female. And most of the patients, around 80% of patients have reported with the pain abdomen for the gastric cancer cases, and 72% have reported with the heartburn for the gastric cancer cases. We did the hematological assays, and we found that in gastric cancer cases, patients was having hemoglobin slightly less, significantly less as compared to the controls. This may be due to the nutrition factors, or various factors may have been associated with the gastric cancer cases. And we did the clinical parameters. In the clinical parameters, we have found that for gastric cancer cases, calcium was significantly less as compared to the control groups. Then we did the ADMA and SDMA estimation by HPLC, and coming to our results, ADMA concentration was significantly less as compared to the control groups, and SDMA concentration was significantly less as compared to the control groups. When we have calculated the ratio, ADM and SDMA ratio, we found that ADM-SDMA ratio was significantly less than the control, healthy control, while there was no correlation in disease control and healthy control groups in the ADM-SDMA levels ratio. So we could focus on this ratio to check the assessment of gastric cancer cases. And for the nitric oxide level estimation, we found that for the gastric cancer cases, this nitric oxide was more as compared in gastric cancer cases as compared to the healthy control. It was significantly higher in gastric cancer cases. And when we have correlated the ADMA and nitric oxide concentration, we found that there is a positive correlation of ADMA concentration and the nitric oxide concentration in gastric cancer cases. And in all these three groups, though there was correlation, but positive correlation was found in gastric cancer cases only. Then we did the DDH estimation, and it was found that DDH was significantly higher in gastric cancer cases as compared to the controls. Then for the mRNA expression of our genes Claudin 7, this is the amplification plot of the Claudin 7. We did the mRNA expression by cyber chemistry, and this is for Claudin 18. And coming to results, we found that Claudin 7 and Claudin 18 expression was downregulated in gastric cancer cases as compared to the controls. And PDL1 we have done, we found that PDL expression was upregulated in gastric cancer cases as compared to control. Then we have checked the mitochondrial DNA and nuclear DNA. Mitochondrial DNA copy number, we found that mitochondrial DNA copy number was less in gastric cancer cases as compared to the control group. Then MMP7 was done in serum level, and MMP7 mRNA expression has been done, and it was found that in both they have both have the symmetry. Level was higher in gastric cancer cases, and it was overexpressed in the mRNA expression also. So we have concluded that ADMA concentration was decreased in gastric cancer cases. It may it was unable to block this enzyme nitric oxide synthase. As a result, nitric oxide stimulation was there, and it increases in the circulation, and which affects on the calcium channel, which goes to the mitochondria and affects the mitochondrial DNA copy number, which may lead to the gastric cancer cases by following the pathway of the dysplasia. And this Claudin gene is downregulated, maybe due to this gene downregulation. This may have the effect on the extracellular protein, this MMP7, and as a result of this, may proliferation is there for the gastric cancer, and there is PDL1 overexpression is there, so that this may have the activated immune response, and due to this activated immune response, it may be helpful in the cancer cases. So we can find that ADM levels is regulated in the dysregulation of nitric oxide production, maybe for gastric cancer, and it might participate in occurrence and development of gastric cancer cases. And PDL1 expression and mitochondrial DNA copy number may be the potential biomarker to identify higher risk of gastric cancer cases.
Thank you, Priyatma. Okay. So, other than the nitric oxide toxicity and the pathway which is involved, is there any other mechanism is known for the gastric cancer?
Yes, so various mechanisms are there. Like we did the SODs also. These are also the free radical pathway. And if we're talking about the nitric oxide, just free radical, on the basis of that we can go. Superoxidase pathways and various pathways are there. We can talk about the apoptotic pathway also. For mitosis, so cascade pathway is also there. Then for today nowadays, PDL1 is having more importance over in this.
We are a very good presentation. See, you have some inclusion, exclusion criteria. In the exclusion criteria, you said smoking.
So smoking and
does it cause gastric cancer?
Yes, sir, it has been reported.
Then why did you exclude?
Sir, because we have to check the nail cases of the gastric cancer, and we have to check whether all the today work we are doing, whether it's associated with, because gas already smoking is known fact. So we are focusing on any marker which leads to the gastric cancer.
We should have considered smoking also as one population to prove that it causes cancer, gastric cancer.
Smoking is always reported, sir. Smoking.
Smokers may not agree with
But
Thank you.
It's really a good question. And you are talking about the whole linkages.
yes, sir.
In one line, what do you say? There's so many things to talk about.
Sir, actually, all the link I have, we have planned in such a way that it should be linked as a pathway, because we are focusing when we are studying something. Only one pathway is there, one pathway is there.
But when you're talking about the biomarker, how you will do that?
That's why I focus on the association, whether this my genes are, whether they are associated with each other. Like I did the clouding, then whether this is. So I have
only one gene which can be taken as a biomarker, because biomarker cannot be.
all the genes is not possible in such a short.
But this is a good presentation.
Okay, thank you, Priya.
Thank you, Dr. Priyatma. Before calling on the next presenter, I would like to, just clear the rules. The marks are given accordingly. So it's 8 minutes for presentation and 2 minutes for discussion part. When the 8 minutes is complete, the bell will be, will ring the bell, and you have to conclude it at around 8. And after that, 2 minutes of discussion. Then we'll again press the bell. So now I would request Dr. Suman Mishra, SGPGI Lucknow, please come and present her talk.
Thank you. Good evening, all of you, and thank you for giving me such opportunities. So today I'm going to present my work entitled Exosomes Secreted by Umbilical Cord Blood Derived Mesenchymal Stem Cells Attenuated Diabetes in Mice. So diabetes needs no introduction because it is a major growing health problem that has been affected worldwide, and in India the proportional mortality is 2%. Diabetes is a chronic disease marked by high level of sugar in the blood, and there are two types of diabetes: diabetes 1, type 1, and type 2. Type 1 is due to destruction of beta cells, and type 2 due to dysfunction of beta cells. And as everyone knows, diabetes causes multiple complications like stroke, heart attack, glaucoma, cataracts. So we are focusing on this part on type 1 diabetes, which is substantial beta cell loss. So in this part, we are trying to how we treat beta cell or regenerate the pancreas. So beta cell replacement therapy is one alternative to administration of exogenous insulin. However, we try to compare the mesenchymal stem cell therapy, which is a recent innovative approach in diabetes, but MSC administration for therapeutic purpose may expose individual to the side effects to cell-based therapy. So we hypothesize first in this part of work to compare the MSCs and MSC-derived exosomes, and to check by inspired to parakine hypothesis, we try to focus on MSC-derived extracellular vesicles-based therapy of type 1 diabetes. So our aim to this study is to understand the therapeutic effect of exosomes secreted by umbilical cord blood derived MSC on pancreatic repair and MSC exosomes on pancreatic repair or regeneration. What exosomes are, basically they are type of extracellular vesicles that contain constituents of the cell that secrete them. So what we plan is in a 3-step process. First, the isolation and characterization of MSC from umbilical blood cord. Second, the isolation of exosomes, and third, to check the effect of these exosomes on MSC and MSC exosomes in vivo. So in these slides, I'm presenting here the successful isolation and characterization of MSC, and which is finally concluded by fluorescence markers of MSC, CD19, CD73, CD105, and which is again rechecked by FACS analysis. And we checked the functional status of these isolated MSCs, and we confirmed their expected tri-lineage differentiation. Further, at step 2, we isolated exosome from MSC, and as we know, the isolated MSC were 132, nearly 132 nanometer in their size and with their characteristic spheroidal cup shape. And they show the presence of exosomal specific marker TCG1, CD3, 63 and CD81. In this slide, we try to check whether MSC and MSC derived exosome share same microRNA profile or not, and we found that they share nearly 90% same mRNA profile. So we move further to check that functional status of exosomes, which is derived from MSC, and we found that exosomes share the same immunosuppressive property as MSC exons. So at the end of these two steps, we concluded that we can use exosome as an alternative to MSC also. So we plan this to translate this or use this MSC and MSC exo in vivo mice model, which is a diabetic mice model. So from 0 to 5 days, we treat STJ for 40 mg per kg body weight, and then up to 10 days, we observe their blood glucose and body weight, and then we treat them up to 7 days with the MSC and MSC derived exosomes, and then we sacrifice mice and collect all the tissue for blood glucose, pancreatic sectioning, and for NGS and macroanalysis. So first, development of mouse model. This slide shows the induced glucose level and decreased body mass that confirms by the histopathological analysis showing that there's a destruction of isolate of Langerhans. That means the mouse model has been developed properly for diabetes. And when we attenuate, when we treat these mice model with MSC and MSC also, we found that MSC exon MSC treatment given the attenuates the diabetic dependent complications. However, we can clearly see that MSC exon did quite better than MSC. We checked blood glucose level and body weight also, and we found that MSC exon MSC treatment lowers the blood glucose level as compared to the previous. And we checked the proliferation, beta cell proliferation in MSC and MSC exo-treated isolates, and we found the significant upregulation of the BRDO positive cells, which reflect the regeneration of beta cells, and the insulin level is also measured, and which is found upregulated in MSC and MSC exo-treated cells. So we want to check what pathways are upregulated or downregulated or affected by these MSC and MSC exo-treated animals, and we found that most of the pathways are either insulin signaling pathway or regenerating pathway. And we can clearly see that there are some molecules that are genes that are upregulated in MSC exo clearly. One of them are REG, that is regenerating insulin molecules, which helps in regeneration of isolate of Langerhans, and there are two isoform IREG2 and REG3 upregulated significantly in MSC and MSC exo. So in summary, this result reveal the production of pancreatic tissue damage with improved histological structure in mice treated with MSC and MSC exo. And hypoglycemia was also attenuated with these treatment, and we found increased expression of genes with MSC and MSC exo. And in conclusion, these results demonstrate a potential therapeutic role of umbilical cord blood derived MSC and MSC exosomes in diabetes by activating pancreatic isolate intrinsic regenerative abilities. Thank you.
Thank you so much.
I didn't explain, sir, because I think I'm running out of time.
No, you have no time.
I will, sir.
Okay. You use this time to answer our questions.
Thank you. I'll try my best.
Very simple question. Why did you select this topic?
This topic.
Why did you select? What problem did you select?
So actually, exosomes, we are trying to challenge that, and despite using MSCs, we can use MSC exosome also, because they nullify the MSC-induced cell-based negative or side effects. Like sometimes they fail, their immunorejection is there. But if we use exosome, and exosome give all the benefits of MSC, but it reduces the side effects. That's why we thought that we could use in exosome in place of MSC. That's why this is our basic hypothesis.
So any earlier studies indicating this?
Yes, sir, definitely. There is one study.
What is new in this?
The new is we compare the complete profile first in exosome and MSC, and the new is the earlier study is in a bone in a dental pulp, and we are using umbilical cord. And the very new, which makes it different, is the biomarker RAG2, which is not known earlier, that RAG2 is playing a role in regeneration of isotope developments. So, because we continuously find RAG2 and RAG3 in different studies. First, we found it in NGS analysis, then we do it their plot, and we found. And one more study I'm not presenting here, that when we track these exosomes, and we found, okay, these exosomes clearly go to the target site and modulate the regeneration, enhances the regeneration of the capacity. No one earlier showed this kind of result.
I just want to
yes,
add up. What is there which brings that exosome to their target? Why you believe that exosome will go to their target? Why?
So this is a open question now, because everyone, we are also studying why these exosomes, because we do tracking analysis and we inject the exosome through tail vein, and they exactly go to their target site, that means the pancreatic cell. So what we hypothesize that there must be some signaling molecules at their membrane, so that they go at their target site. So this is open question, sir. I'm also studying this. Sorry, ma'am? So, ma'am, it may possible that they go somewhere else, but we checked in pancreatic cells and we found it, their presence in pancreas also. This is also further for what make these exosomes directed towards the. Sometimes I think the cow go inside this has some messenger property, so that they are like they have an address type of thing at their membrane, so that they have to go at their target place also. We are hypothesizing this, and we are focusing on this area, that their membrane may have some address kind of signaling peptide or molecule, so that they have different address where they have to go. I think so.
So, Suman, the exosomes derived from umbilical cord.
Yes,
sir. So it's not necessarily that, okay, even though you have a pethotropism, because you are reducing the diabetes in them. So you have the pethotropism, the target might be into the specific cells. But exosomes, they are derived from umbilical cord, they can also reach into the different regions as well. So what is your control experiment in this?
Sir, in this exosome. Yeah. So actually, this is a piece of work we already did some part in the bone marrow derived exosomes also. So for control, we are also trying. So
the reason why I asked is, how do you know that this is the efficacious method in which you achieve the regeneration of the pancreas different degrees?
Yes, sir.
So for that, until unless you have the parallel controls, it becomes different. But in any case, this is a nice study. Thank you very much.
Thank you.
You can answer that.
Thank you.
Thank you, Dr. Suman. Now, without wasting any time, I would like to request Dr. Meenakshi Sharma from IABS, New Delhi, to present her talk.
Very good evening to all esteemed faculty members and my dear colleagues. First of all, thanks to IABSCON team for giving me opportunity to present my work on dynamic expression of transcription factor OLIG2 and platelet-derived growth factor receptor alpha in white matter following severe traumatic brain injury in humans. So, coming to the introduction, traumatic brain injury is a silent epidemic and major cause of death and disability worldwide. Late onset pathological indications of traumatic brain injury, such as cognition problems and other problems, which are directly related to aberrant axonal functions and results in progressive atrophy in the white matter. So the oligodendrocytes, which are most delicate cells and more vulnerable in the molecular processes and involved in the demyelination post severe traumatic brain injury. The oligocyte progenitor cells such as OLIG2 and PGF alpha subsequently release at the site of injury within 48 hours and overexpresses at least within 2 weeks. So due to the lack of clinically significant treatment strategies and ambiguous scars and inconclusive prognosis of post-severe traumatic brain injury, the study was conducted to examine the role of OLIC2 and PGR-alpha in severe traumatic brain injury patients. Coming to the hypothesis, OLIC2 and PGR-alpha may possibly affect either the white matter atrophy or remyelination in the severe traumatic brain injury. So in the normal condition, these are the oligodendrocytes, and this is the neuronal cell body, and oligodendrocyte helps in myelination of the axon during the acute phase of traumatic brain injury due to the damaged oligodendrocytes, which leads to the demyelination of the axon and later results in the aberrant signal transduction inside the brain. And what happens in the remolding phase after traumatic brain injury? The OLIC2 and PGR-alpha proliferate at the site of injury. They proliferate, migrate, and differentiate to remyelinate the demyelinated axon. So what's the right time and what's the right expression? This is the major question in the remolding phase after traumatic brain injury. So the objective was to investigate the expression pattern of transcription factor OLIC2 and PGR-alpha in white matter after severe traumatic brain injury in comparison to the non-TBI among humans. So coming to the materials and methods, post-mortem brain tissue from the corpus callosum has been collected from severe traumatic brain injury patients, non-traumatic brain injury patients involving without brain, and the control patients without any injury. So the patients whose GCS score was less than or equal to 8 at the time of admission with positive CT findings were included in the study, and the patients who have had post-trauma craniotomy or any other surgical intervention related to brain, neuropsychological or any neurodegenerative diseases, penetrating injury to the head or at oxygen more than 24 hours were excluded from the study. So after collecting the patient demographic and the CT findings, demyelination grading using the HNE and Luxor fast training has been done, and the IHC of myelin basic protein, OLIC2 and PGR-alpha has been done, and the mRNA expression of OLIC2, PGR-alpha and housekeeping gene has been done. So these are the demyelination grading. This is the section from the corpus callosum showing the grade 3 demyelination, reduction in the oligodendrocyte cells. The second section shows the section from the corpus callosum showing grade 4 demyelination. This is the section from the control case showing the normal pathology of the brain. This is again the demyelination grading using the Luxor fast blue fast training and the IHC of myelin basic protein. There's a section showing the demyelination in both the sections, and there is a myelinated axons in the control case. This is the IHC of the OLIC2 showing the cell counts, OLIC2 counts in the severe traumatic brain injury patients compared to the controls. This is the amplification plot for the PGR-alpha showing the MELD curve also. This is the amplification for OLIC2 and the MELD curve. Coming to the results and the demographic and the clinical details, age-wise, we can say the age group from 20 to 29 years, which is the young population, is more vulnerable for the traumatic events. Sex-wise, males were more exposed to trauma comparative to females. And coming to the GCS score, most of the patients were in deep coma, that is the GCS score from 3 to 4. So, for the severe traumatic brain injury and non-traumatic brain injury, the post-mortem interval was 13.5 hours compared to the control, which was 16.8 hours. And the survival time group-wise was, for the severe traumatic brain injury, was 40 hours compared to the non-TBI, which was 1 hour. Coming to the mode of injury, RTA were maximum, followed by fall in severe traumatic brain injury, and which is followed by assault in non-traumatic brain injury. Under the fall category, the fall of a height more than 10 feet is maximum compared to the simple falls. According to the type of collision, rear impacts were maximum, followed by side impact in severe traumatic brain injury, compared to the non-TBI, where the side impacts were maximum as compared to severe TBI. Coming to the CT findings, 79.3% were hemorrhage, followed by subdural hematoma, contusions, midline shift, basal systems open. Coming, what we do, like CT findings were later classified according to the Rotten CT classification. So there is a significant difference between the severe traumatic brain injury and the non-traumatic brain injury. And clinical DI score was found to be non-significant among the two groups using the Adams classification. This is a demyelination grading group-wise using the HNE grading and Luxor fast group grading. So in the severe TBI patients, we found a significant difference as compared to the non-traumatic brain injury and the controls in both the type of gradings. And coming to the demyelination grading according to survival time, what we found, like estimation, we found like in the HE group, the demyelination grading increases with time, but in the Luxor, we found the higher grades in days more than 3 to 10 days. This is the IHC of OLIC2 cells in different groups. So we found a significant difference group wise, and in the survival time, we found a increase in the number of cells with days. This is a group-wide relative expression of the OLIC2 and PGF-alpha in white matter. We found a significant difference in the severe traumatic brain injury compared to the non-TBI and the controls. This is the relative gene expression of the OLIC2 and PGF-alpha after severe traumatic brain injury according to survival time. There is a steady increase at around 80 hours in the OLIC2 compared to PGF-alpha, and at 150 to 200 hours, like around 6 days or 3 to 10 days, we can say, both the genes are tried to equilibrate themselves. So, and this stuff reminds more of action potential of a membrane during the state of depolarized state, just like in traumatic brain injury. This is a schematic illustration of an oligodendrocyte neuronal unit, where when there is an uninjured node, there are various kind of pumps are present: sodium channel exchangers pump, ATP dependent calcium pump, sodium channel pumps, and inside the membrane there are microtubules and other neurofilaments. And when there is a traumatic axonal injury, what happens? Due to the axonal perforation, there is a uncontrolled flux of these calcium ions inside the membranes, and due to this there is a depolarization, and which later results in axonal varicosities, axonal swellings, axonal retraction bulge. Okay, just 2 more minutes. Okay. So after what happens, like after closed TBI, these cells try to increase in size, and try to migrate and proliferate and try to demyelinate the demyelinated vessel. So during STBI at 6 to 24 hours, these cell increases. Later at 3 days, the again increases, and at 3 to 10 days, we can say this is the most critical phase, and this is the right time for therapeutic intervention, so that the cells will proliferate, differentiate, and either to remyelinate or demyelinate the lesion. So this is a summary of my demographic results. And coming to the finding and conclusion, higher demyelination rates were observed between 3 to 10 days. There is a significant difference of expression pattern of both OLIC2 and PGF-alpha and severe TBI compared to the non-TBI and controls. Both the genes are OLIC2 is increased at 80 hours, and PGD2 increases after severe TBI, well lags behind the OLIC2 gene at 150 to 200, and try to equilibrate. So we can say this is a 3 to 10 days, maybe the most critical phase and appropriate time to assess the recovery, and this is the most appropriate time for therapeutic intervention, we can say. So this opens this, we can say both the markers are the key factor in reaction to glial cells or to brain injury, and this study it may direct towards the right therapeutic target intervention time after severe TBI, may further contribute to design the clinical significant treatment strategies. Thank you. Thank you so much.
Thank you, Vinay. So now, based on the expression, these are the natural phenomena when it comes to the traumatic brain injury. Severe, moderate, or you have the mild form of the traumatic brain injuries. You do have these expressions of these two. So either you take the oligo2 or the platelet-derived protein factors, the expression patterns they change. And also based on the severity and also the duration in which the traumatic brain injury is caused. So accordingly, this is a dynamic phenomenon. Based on your results, so what is the implication when it comes to the evolving the strategy for the treatment of this traumatic brain injury?
We can suggest a panel of markers also, and
there are multiple factors.
Okay, and
multiple markers are there. So based
why do you choose these molecules can be suggested so that there is a progression of these precursor cells at the site of injury, and they are there should be there is a progression for these precursor cells so that there is a natural inducive neurogenesis of the brain. We can suggest some anti-suppressor molecules.
That happens much later because until unless the brain gets stabilizes, following the traumatic brain injury.
We can think of human OPC's transplantation so that there is a differentiation.
Okay.
Think of
the temporal scales are extremely important when we talk about injury.
You might have taken the immunoethics committee approval.
Sorry, yes, sir, yes, sir. Yes, sir. The study is also coming from ICMR also.
There will be many other treatment protocols at the time of such an injury. So
Sir, there is like still like neurosurgeons are fighting. So, what is the right time for doing the craniotomy or cysternectomy? So, what's the technique? I am working in trauma center from the last many years. So, what I saw, like what they did, like there is no time actually. They decide whether at what time they do the surgery and which type of surgery should be the right time. Either craniotomy or cysternectomy should be done, in which population it should be done, and they just remove the flap to release the intracranial pressure. That's it. So like I'm thinking like being working in trauma center, so for the neuro trauma patients should be like that's why I'm like
can guide them. Your work can
yes sir. For the clinicians and also like
what kind of approach they have to make.
Like I have to like further do my.
No, it's really interesting and tough work.
Essentially, you need to understand the pathophysiology associated with dynamicity in the traumatic brain injury. Whether these studies can translate in terms of the intervention in the later stage. Yeah,
because just people are still working in like animals and all, and working took a complex brain of human. So what?
Keep it up.
Thank you so much.
It's very much needed.
Thank you.
Okay. Hello, Dr. Priyadarshini, can you hear us?
Yes, sir. Am I audible, sir?
Yeah, you're audible. You can start sharing your screen.
Yes, sir. Sir, is my slide visible?
Yes. Yes, it is visible.
Can I start, sir?
Yes, go ahead.
Good evening, everyone. I am Priyadarshini, PhD scholar from the Department of Biochemistry, Chitpur. Today, I am going to present my work entitled Association of Variation in the Vitamin D Receptor Gene with Chronic Kidney Disease Risk in South Indian Tamils. Coming to introduction, chronic kidney disease is a condition in which kidney structure deteriorates and causes a progressive renal function decline. The two main consequences in CKD is disruption of mineral and bone metabolism, which is followed by the vascular calcification. Vitamin D plays an important role in the management of bone health, and it also helps in the reducing CKD risk by inhibiting two important pathways. It's a renin-angiotensin pathway and NF-kappa B pathway, which leads to vasoconstriction, fibrogenesis, inflammation, renal injury, and renal failure. Since vitamin D receptor mediates the genomic effect of vitamin D, in this study, we wanted to investigate the relationship between the variants of vitamin D receptor gene and susceptibility to CKD in South Indian population. We have chosen 4 single nucleotide polymorphisms from the vitamin D receptor gene. These are the functional consequences of the SNPs we choose. The first SNP is RS2228570. It's present in the 5' end of the exon near promoter, which leads to the altered start codon in the VDR gene, leading to a longer VDR protein with less transcriptional activity. And the other 3 SNPs, RS154410, RS7975232, and RS731236, regulate the gene expression of this vitamin D receptor through the alteration of mRNA stability and disruption of splice sites for transcription. All these polymorphisms affects the vitamin D receptor activity and subsequently vitamin D mediated effects. It's a cross-sectional study with the South Indian pre-dialysis non-diabetic CKD cases between the age of 18 to 70 years and age, gender, and ethnicity matched healthy controls. The participants who are pregnant, having active infection, and having pre-existing atherosclerotic vascular disease, malignancies, and diabetes mellitus were excluded from the study. The sample size for the study was calculated using CATS Power Calculator software, and the final sample size was 408 patients with 204 participants in each group. Coming to the methods, we collected 5 ml blood samples from 75 CKD patients and 150 healthy controls, and the fasting blood glucose, lipid profile, urea, creatinine, calcium, phosphorus were taken from the case records. And the disease severity of chronic kidney disease is calculated using chronic kidney disease epidemiological formula. And the DNA extraction was done using commercially available kits, and the DNA was quantified using nanodrop. And the SNP genotyping was done by Tachman 5' allele discrimination assay by qualitative PCR. Coming to statistical analysis, Normality of our data was assessed by Kolmogorov-Smirnov test, and our data was expressed as mean with standard deviation or median with interquartile range. The observed allele frequency in cases and control were compared with the expected frequency to check the Hardy-Weinberg equilibrium by chi-square test, and direct gene counting was done to calculate the allele frequencies, and odds ratio 95% confidence interval was calculated using chi-square test, and haplotype frequency and linkage disequilibrium within the SNPs were measured with the HaploView software, and the p-value less than 0.05 was considered significant. Coming to the results, these are the demographic and clinical characteristics between the CKD cases and controls. And this is the routine biochemical parameters of CKD cases. Coming to the single nucleotide polymorphism results, this is the first SNP RS154410. When we checked the genotype and the allele frequency of this SNP, we didn't find any significant difference between the cases and control. This was confirmed using the genetic model analysis, where still we didn't find any difference. This SNP doesn't have any influence on the genotype on the phenotype of the CKD. This is our second SNP RS731236. When we checked for the genotype and allele frequency, still in this SNP also we didn't find any significant difference, which was also confirmed using genetic model analysis. This SNP also doesn't have any influence on the phenotype of CKD. Coming to the third SNP RS228570, we found that the AG genotype and the A allele was significantly different between cases and control, and it's shown to have a protective role against the CKD disease. This was also confirmed using the genetic model analysis, where AG plus AA model and the AA allele was found to be significant between cases and control, and here also it showed a protective role. Still, this SNP doesn't have any influence on the phenotype of CKD. This is the result of our fourth SNP RS7975232. We found that the CC genotype and C allele of this SNP was found to be significantly different between the cases and control, and it showed to have B and risk allele for CKD. This was also confirmed using the genetic model analysis, where the CC allele was found to be significantly different between cases and control, and here also it proved the risk for the CKD disease. Still, this SNP doesn't have any influence on the phenotype of CKD. When we checked for the linkage disequilibrium between these SNPs, we found that a strong linkage disequilibrium was observed between the second and the third SNP, and moderate linkage equilibrium was found between first and second and first and third SNP, and weak linkage disequilibrium was found between the first and fourth SNP. When we checked for the haplotype frequency of these SNPs, we didn't observe any significant difference in the distribution of haplotypes between the CKD cases and controls. From our study, what we found is there was a significant association between the RS7975232 polymorphism with CKD susceptibility in South Indian population, whereas the other polymorphisms failed to contribute to the disease risk. The observed risk C allele in this study was in concordance with the previous study related to CKD and in Chinese and Indian population. This SNP was also found to be associated with osteoporotic risk in the postmenopausal women.
Dr. Priyadarshini, sorry to interrupt. Your 8 minutes are over. Please conclude.
Yes, sir.
Conclusion is that.
Yes. Our study indicated that this SNP CC variant could be a risk allele for the CKD patient in South Indian population. We studied only a few SNPs, which could be a limitation, and since it is an ongoing project, sample size was not fully achieved. Thank you for JIPMER for providing the funding for this project. These are my references. Thank you.
Sir, any queries?
Why did you select only Tamilians? Tamil Nadu?
Yeah, sir. It's a Tamil population, sir.
Why? Why? Anything special?
No, sir. Actually, based on the ethnicity, all these SNPs will differ, sir.
Why is it the question
it won't
of
be.
convenience? Is it a question of convenience getting the study population easily? No? You can't get North Indians in Pondicherry.
Yeah, sir. Actually, it
What is
will
the
be
real
some
logic?
population.
What is your logic to choose only this?
this population?
Sir, I couldn't hear properly. Sir.
What is your logic to choose only 7 million?
Sir, it may differ. Actually, we are in Puducherry and patients, maximum patient load we will get from Tamil population. So we choose to maximum.
No, second thing is SNPs also vary. Is that right? With the populations?
Yeah, sir. With the population, it will vary.
There's nothing wrong in doing something.
So, Priyadarshini, what is the significance and the translational aspect of your study when we talk about chronic kidney disease?
Sir, actually, this is a little part of my study. We are still following up those CKD patients, and we will follow them for the CVD risk and for the bone-related complications, sir. Based on that, we are planning to develop an algorithm which can help us to predict in the prior stage so that the treatment can be given for the patient in the early stages, sir.
Based on your SNP studies, you are going to have say early prognosis or anything?
Yes, sir. Actually, not only based on the SNP, sir. We are also planning to do some protein markers evaluation with all those and some scans for bone health and the CVD markers. Based on this, we are planning to develop an algorithm, sir. Thank you, sir.
Okay, thank you, Dr. Priyadarshini. Now I would request Dr. Upinder Singh from IABS, New Delhi, to please come and present the talk.
Good evening, faculty, teachers, and all the my colleagues. The topic of my presentation is potential role of lactic oxygen species and signaling of NRF2 gene and its expression in polycystic ovarian syndrome. PCOS is the most common endocrine metabolic disorder affecting up to 6 to 20% women of reproductive age group in our country, and clinically PCOS delineated variety of phenotypes including chronic anovulation, hyperandrogenism, insulin resistance, obesity, anxiety, and infertility. So a little bit about reactive oxygen species. These are frequently exposed in cellular compartments and also involved in many diseases including cardiovascular diseases, diabetes, recurrent pregnancy losses, preeclampsia, intrauterine growth restriction, embryonic disruption, and even fetal distress, leading to death. So, mechanism of reactive oxygen species in PCOS patients. Excessive generation of free radical affect directly or indirectly to cellular components of protective role, or excessive generation of ROS that not only cause mutation but also induce irreversible modification of proteins, lipids, and advanced glycated end products in production of severity of diseases of cell death. So the present study was planned to determine the level of oxidative stress markers like prolidase, superoxide dismutase, malondialdehyde, ferric reducing antioxidant in plasma, and also NRF2 gene expression in to understand the pathobiology of PCOS patients. So it was hypothesized that evaluation of oxidative stress and expression of NRF2 gene in relation to metabolic disorders with the irregular menstrual cycles and more menstrual pains in the PCOS. Objectives were to determine the expression of NRF2 gene in PCOS and healthy subjects, and to determine the activity of all these oxidative stress markers in PCOS and healthy subjects, and also to correlate these stress markers patient versus healthy control. So it was a prospective cross-sectional study. Ethical approval was taken from the IABS Ethical Committee. So to define PCOS patients, that Rotterdam guidelines were taken under consideration, and according to that, at least 2 of the 3 parameters should be present.
First one is oligomenorrhea or anovulatory cycles with menstrual irregularities. Second is hyperandrogenism. Third is ultrasound evidence of polycystic ovarian disease. It may be transvaginal or transabdominal. So there are 4 different phenotypes of PCOS have been classified, A, B, C, and D. Among these, type A is most severe form as it includes all the above 3 parameters. But type B is the most common. So for study, my patient population was divided into group 1 PCOS, and group 2 is healthy control. Among PCOS population, total 28 patients screened and 1 refused to participate and 1 not met with the criteria. So 26 patients, and like in group 2 healthy controls, among 30 only 22 were recruited. So these were inclusion criteria and exclusion criteria for both the group of patients. In group 1, inclusion criteria was according to Rotterdam criteria. Secondly, those patients willing to participate in the study. And the exclusion criteria for group patients were, group 1 was pregnant subjects or desiring of pregnancy and history suggestive of any systemic illness like coronary artery disease, diabetes, Cushing syndrome, and other endocrine disorders. And also patients having known history of drug intake like androgens, anti-epileptic drugs, steroids, and other like vitamin D, metabolism drugs. So last was the subject who refused to participate or lack of informed consent. And in group 2, the healthy controls were normal healthy subjects aged from 18 to 35 years of age and willing to participate, not on any antioxidant therapy and having good physical health. And those were excluded like with severe comorbidities, past history of heart disease, other systemic disorders, and also those having history of smoking, alcoholism, and also on NSAID in the last 3 months. So this figure shows the type of frequency distribution of PCOS. Like we can see phenotype B was most common. So on this box plot shows the levels of ferric reducing ability of plasma in PCOS versus non-PCOS. So we can see that here the level of FRAP are significantly lower as in the PCOS as compared to non-PCOS. And this plot shows the levels of malondialdehyde levels in the PCOS versus non-PCOS. So MDA is higher in PCOS patients, so it's a significant difference. And this shows that the level of superoxide dismutase in PCOS versus non-PCOS. This box plot shows also significant difference between PCOS and non-PCOS patients. And this box plot shows prolidase levels in PCOS versus non-PCOS, and there was significant difference between both the groups. So finally, there was relative expression of NRF2 gene mRNA in the both groups, and here it was seen that there was significant lower values of the NRF2 gene expression in PCOS as compared to healthy groups. So this table shows the result of all the oxidative stress markers. We can see all the value differences were significant. So conclusion of our study was the decreased level of superoxide dismutase indicate that there may be synergistic interaction of superoxide radicals and increased level of liquid peroxidation that may be important determinant to assay the severity of this disease. And increased level of prolidase activity could be the RS associated phenotypic marker that may help in defining the phenotype in PCOS. And also the suppression of NRF2 gene could be a novel prognostic therapeutic biomarker in PCOS. And there were a deranged level of SOD, lipid peroxidation, prolidase enzyme could be oxidative stress markers in the PCOS. So, and to correlate decreased level of FRAP and SOD in the suppression of NRF2 gene that may be contribute to the severity of PCOS. And increased prolidase activity and downregulation of NRF2 gene contribute as a novel approach to provide a better understanding of pathophysiological mechanism involved in PCOS patient. So there is overall assessment of NRF2 gene expression may help in formulation of therapeutic strategies along with the targeting of defined phenotype, it may be the future plan to treat PCOS patients.
Thank you, sir. Is enough to be so specific to the PCOS?
Sir, no, it is not only specific to PCOS patients. It is related to other diseases also, but its expression is seems very lower in PCOS patients if you compare with the healthy subjects.
Because you said that you are going to use this as a biomarker.
Yes, sir. So this is the going in a cell process. It can be used as.
It can be used as.
So, what was the rationale to take NRF2, FRED, and SOD,
Yes,
FDA? Why you choose? And you don't correlated it in your study. So, why you choose different markers? What was the rationale?
No, sir, I took an oxidative stress markers like SOD, FRED, and polydase.
But there are a lot of oxidative markers, and also, did you use SOD 1 or 2 or both of them? So, said you showed SOD. Then
yes, sir.
Which SOD it was?
Sir, SOD 1.
1. So, 1 is a cellular SOD or mitochondrial SOD? What is it? Is
Sir, cellular.
mostly not. It is always cellular. Okay. So, SOD2 is a mitochondrial. Okay.
So many markers you have selected.
Yes, sir.
Which one you recommend? Economically feasible, they are common one.
So polydase can be used.
Cost.
So, but yes, right now it is not cost effective.
Or population should afford it.
Yes, sir.
And laboratory medicine is commercialized.
Yes.
How common man can afford.
Yes. Scientific process.
Okay, thank you.
Okay, thank you, Dr. Upender. And now we have the last presenter. And now I would like to request Dr. Shruti CR from Thiruvananthapuram to please come and present her talk.
A very good evening to all. My name is Shruti CR and I am a PhD student and working under Dr. K.G. Raghu at CSIR NAIST Trivandrum. I would like to thank the IABS organizing committee for giving me this opportunity to present my topic, involvement of middle lifestyle in cancer in occurrence in diabetes. According to IDF Atlas, in 2020-21, approximately 537 million people were living with diabetes, and almost 6.7 million people deaths were reported in 2020-21. Even more scary thing is that link between the cancer and diabetes that has been established recently. And the common risk factors include age, sedentary lifestyle, food habits, etc. Now, what is mealyl lifestyle? Mealyl lifestyle is a byproduct of glycolysis that has seemed to be increased in the case of diabetic patients. It is a dicarbonyl compound that is highly reactive, almost 20,000 more times reactive than any other reducing sugars that can form advanced glycation end products. So it is the most potent precursor for advanced glycation end product formation. And recently it has also been implicated in many types of cancer. Factors contributing to the accumulation of mealyl lifestyle. Mealyl lifestyle is formed mainly during the hypoglycemic conditions, that is the hallmark of diabetes. And some other factors that contribute to the mealyl lifestyle formation include inflammation, oxidative stress, etc. Once these mealyl lifestyle are formed in the body, they can react with your proteins or lipids or DNA to form advanced glycation end products that can bind with the receptors for advanced glycation end products and start a signaling pathway, the pathophysiological signaling pathways that can include inflammation, thrombosis, apoptosis, etc. Now, how is the mineral cell formed in our body? The main contribution of mineral cell formation is done by the glycolysis. Here, the glycerol 3-phosphate and the DHAP, the intermediates of glycolysis, can be degraded non-enzymatically to form mineral cell. Other pathways include glucose auto-oxidation, acetone-threonine catabolism, lipid peroxidation, etc. So, as I told you, this is a very toxic compound. So, our body has its own defense mechanism. So it can be detoxified by inside our body by several enzyme systems. The main one is the glycolysis system, which includes two enzymes, glycolysis 1 and glycolysis 2. And the other enzymes like aldehyde dehydrogenase, aldose reductase, and DJ1 are the other enzymes. Well, as I told you, mineral cell is formed and detoxified in our body. Once there is an imbalance in our body during the pathophysiological conditions, it can react with your proteins to form AGs or advanced glycation end products. And these are the major players of the major diabetic complications during the diabetes. Our major objectives involve the effect of mineral cell on the age formation, various receptors, and glucose metabolism. The methodology used here was 50 micromolar concentration of mineral cell were subjected to the in vitro system G2 cell lines for 24 hours, and these cells were then subjected to various biochemical and protein expression studies, mainly involved in glucose metabolism. Here we have used aminoguanidine as the positive control. Coming to the results, mineral cell seemed to be non-toxic up to a concentration of 500 micromolar, but at 1 millimolar, it reduced the cell viability. And we have selected 50 micromolar for our studies, at which it has produced a lot of loss in the G2 cell lines. Effect of mineral cell and glycolysis system. When we treated the G2 cell lines with glycolysis with the mineral cell, it severely downregulated the expressions of the detoxifying enzymes GLO1 and GLO2. It also increased the production of mineral cell hydroxyimidazolones or MTHs. These are the major advanced glycation end products formed by mineral cell. It also increased the expression of receptor for advanced glycation end product in G2 cell lines. The most surprising result that we got in this study was the increase in the glucose uptake. We were expecting a totally opposite result, as the most of the toxic compounds reduces the glucose uptake. But in our study, we found that it is increasing the glucose uptake. It also increased the GLUT transporters like GLUT2 and GLUT1. And we also, we wanted to know where this glucose is going. So we checked the enzymes for glycolysis like HK1, PHK1, and amylase-1, and we found that the expression of these proteins are also increased. Now, for a brief introduction, I would like to tell you the cancer progression and Warburg effect. Cancer progression mainly depends on activation of mutations in the oncogenes and mutations on the tumor suppressor genes and the modulation of the microenvironment of cancer cells. And here you can see at normal cells, the glucose uptake is normal and the pyruvate is directed towards TCA cycle. But during the cancer cells, Warburg effect, glucose uptake is increased and the pyruvate formed here is directed towards lactate production. And we checked whether the pyruvate formed through the upregulated glycolysis is being converted into lactate or not. It was found that there was an increase in the lactate production along with the enzyme lactate dehydrogenase that converts lactate pyruvate to lactate, and there was an increase in expression of the inhibitor protein of the PDH that converts pyruvate to acetyl-CoA. These are the major players, proteins involved in the metabolic disturbances in the middle acyl-treated cells. One of the most important protein are HIF1 alpha and CM/C that can actually increase the glycolytic pathway as well as the growth of the cancer cells. Mostly cancer cells are subjected to this aerobic lipolysis because of the non-functional or dysfunctional mitochondria. And we have seen that there was a decrease in the mitochondrial mass as well as the proteins expressed for biogenesis of mitochondria. We have also seen that the oxygen consumption rate was decreased, which explains the presence of hypoxia-inducible factor. The summary, middle acyl increased the ROS production, it increased the RAGE expression, it also upregulated the glycolysis, and there was a aberrant glucose metabolism that was seen in the G2 cells treated with methyl. My acknowledgement goes to CSI, Director CSI Anist, my supervisor Dr. K.G. Raghu, ACSI New Delhi, and UGC for financial support. Thank you.
So, Shruti, how do you translate your work? Is treating the diabetes.
So, middle acyl diabetes is a very complicated disease. We don't have a proper target for this disease till now. So, middle acyl can be targeted as can be subjected as a target against it. It is an emerging biomarker also. We can develop more and more scavengers for it. Like the aminoquinidine I have told you, but it has severe clinical complications. So it is not a drug yet.
Good. Good presentation. What are the external sources of energy?
Energy. Food is there, sir. Mostly the
coffee. South Indians love coffee.
Coffee is there. Honey is there. Honey is there.
Stop drinking coffee.
We can reduce the ingestion because most of the time the middle acyl or the advanced glycation end products are only 10% percent of the ingested things are absorbed by our body. So we can reduce the ingestion of these things. Mostly we can avoid. The half-life of middle acyl is it is like is it quite stable? It is stable. That is why we need the enzymes to degrade it. It's spontaneous. It is generated spontaneously without the presence of enzymes. But once it's formed, the enzymes are needed to degrade it.
Okay, thank you, Shruti. Thank you.
So with the presentation of Dr. Shruti Sia, we have come to conclude this session. But before we conclude, there is an announcement that the award for this session will be announced in the convocation only. So you all are requested to attend the convocation. And before we conclude, let us facilitate our esteemed chairpersons. So I would like to request now.
Secondly, first one, announce the first one that we request SM Hadi sir.
Sir, Professor Hadi sir, please
come in.
Come on. Yeah. So I would like to request Professor Tupil Venkatesh to please come on to the moment.
All three of us will come. We can give it a percentage.
That is awesome. And now I would like to request Kutela sir to please come. to facilitate B.S. Rao sir. B.S. Rao sir.
Sir, sir, थोड़ा उस side
Now, I will please request Bipin Tiwari sir to please come on to facilitate. Dr. Professor Goel sir.
Sir, please.
Thank you.
So with this, we have come to the conclusion of this session, and we will meet at 7 p.m. What? Yes, convocation I have already announced. So 7 p.m. for convocation. Yes, you can even come before. That's all. Thank you very much.
शेड्यूल में चेंज
फाइनल करके देखिए रिकॉर्ड पर यूट्यूब
Whether they have spent that much time in the lab. They have independently developing
to guide us. And also, overall, what is the object? Girls have understood whether they have overall understanding of the study. Don't wait for every
अरे क्या कहते हैं अंदर ब्रो यानी मशीन वहां लगी ग्राउंड में रिसर्च लगाएंगे
I'm putting I'm coming, sir. You don't leave. This is your confidential stuff, and these things will go, and these files you can take and the files. Thank you, sir. Thank you very much. No time to go home, do what no, there's no time. We really go here. There's nothing to get ready. Once we put the goal, everything will cover. Yes.
You answer your own question. You answer your own question.
நீயே தூங்கலையா அவ்வளவு நேரம் குழந்தைக்கு சுத்தியே தானே இந்த தூங்க வைக்க தானே அங்கெல்லாம் தூங்க சரி இது பண்ணுவேன்னு சொல்லிட்டு கொண்ட போட்டுருவோம். இந்த பக்கம் சின்ன இந்த பக்கம் இல்ல இல்லையா இங்க சின்ன தூக்கி வரணுமா தூக்கிட்டு வரணுமா தரோமா Popsi ama. My like சார் அவங்க பாட்டி தானா நான் குடுக்குறேன்
they have to leave as per the speech by the General Secretary. So I welcome all the delegates, and I feel that all the delegates will have the comfortable stay, and the organizers has arranged all the facilities for the stay of the delegates, and they are doing their level best to give the local transport facility and all the facilities to the delegates. And I wish a great success for this conference. Thank you.
Thank you, sir. Now I request Dr. Hari Sharma, Founder President IABS, to address the gathering.
Good evening to all of you, my friends and dignitaries on the dais, audience with the respected professors, students, and also the fellow of IABS. I'm very delighted to be here. In the last 10 years, the academy, which was a little kind of thought which we were trying to propagate, had a discussion together with my best friend Abbas Ali Mehdi, and we thought, let's start something which will become a platform, and this platform has a simple motive: just friendship. We want to create a friendship between clinicians and scientists, and this was the motive day one we thought to propagate and created this academy. Now we are seeing the 10 years down the line that we have done relatively well. We have established several awards. We have the fellows program started, but at the same time, this platform remained exactly the same motive of creating friendship in such a way that they can do better research, they can develop better therapeutics and also management of patients. Simply because if they come together, the clinicians, those who are working day in, day out, as well as the scientists, those who are working in the lab, if they are friends, they can really develop enormous innovative ideas together. And those innovative ideas, we have seen it today. Also, one of the young boys was showing new tools and technology in the AI, in the artificial intelligence. I think our academy is a broader horizon. Biomedical is a term which covers the entire human spectrum of the disease and well-being. It has thematic chapters all over the country. We conduct every year one annual conference, which is international, but also almost a dozen local chapters. They organize their own events, workshops, or CMEs in different cities, from Srinagar to Trivandrum, which we have already witnessed over the last 10 years. I think what I would like to just convey that we keep this, this little seed which we sowed 10 years ago has come up from the ground, that it can become slowly a small plant to the tree. And that is, a kind of dream come true for me as well as for my other colleagues and friends, those who are really, dedicated to this academy. So with these few words, I would like to welcome all of you. Would like to congratulate both the centers, the entire working team from the KGMU, and also the enormous facilities and amazing hospitality from IRA University, and dynamic leadership of Farjana and Abbas Bhai. I mean, I have no words to really convey that this is amazing setup. And we all cherish these, hospitality and the way they really conduct the things. So I'm very delighted to be here. I come for every conference I have been there, but at the same time, I would like to wish and bring more and more the young minds joining us, more and more people, those who are able to contribute with their experience, with their research and theoretical experience, surgeons and the clinician, those who are seeing the patients. If they can, just give their advice, they come up together with the scientists, those who are doing day in, day out the research. I think that is the key. If we can develop much more, and then that's how the better therapy and innovative ideas will develop. So with these few words, once again, welcome to all of you, and thank you very much.
Thank you, sir. Now I request Professor Tupil Venkatesh, pattern IABS, to say a few words of wisdom.
Very happy and healthy evening to all of you. What a great congregation. I am asked to speak few words of wisdom. Who am I? When people with wisdom have assembled here, I should get some wisdom from these people. I am a small man. I can tell you one thing. I don't know whether you noticed. Normally in any university, there's only one vice chancellor. Today, see on this dais how many vice chancellors. How many vice chancellors are present here? This is a congregation of wealth of wisdom. When I say wealth of wisdom, I think we should clap to all of our people with wisdom, including all of you. You are going to clap for yourself. All of us, whether young or old, we have only 6 activities. Please remember, there are only 6 activities. In these 6 activities, we don't have control on 2 activities. Nobody has control. That is birth and death. We have no control. In between, there are 4 activities, madam. We have absolute control. Why I'm telling you, the 4th activity where we have control is very important, where we are all concerned. The first activity after we are born, as a small child, your grandchild, play. Can play good game, fair game, unfair game. This is under the control. We don't allow a child to play unfair game. After that, as the child grows, kindergarten, LKG, UKG, first standard, second standard, medical education, learn. There also there's a control. An university like this where we have assembled teach us only good things. We should learn. That's also under control. Fantastic. Look at Dr. Farzana. Controlling the education system in this place. There's a control. Third, many of you, the next activity, earn. You're earning. You have to earn to support your family in the right royal way. It looks like a philosophical, but it has a deeper meaning. We have to earn under control. Many people earn without control also. Now come to people who have assembled here, people who are getting fellowship, including myself at the age of 76. I tell you, there's one activity where I need to have control. That activity is return back to the society. Whatever we have learned, whatever we have earned should be returned, because we cannot carry anything with us. Even my memory today with you people will not come with me. That also should be returned to right person. This is what IABS is. Look at the vision, mission under the President of Raghu. You are identifying, recognizing. One day I would like to see every one of you on the dash. That should be our aim and goal. All the best, all of you. I am honored. I'm honored to be part of the honorable audience here. Thank you.
Thank you, sir. It is my great pleasure to invite our guest of honor, Dr. Amar Rizvi, to address the gathering.
All the best and honor
Respected Mr. Professor Dr. Raghu, Professor Hari Sharma, and Professor Thupil Venkatesh, respected Churamaniji, former Vice Chancellor, King George Medical University, Professor Dr. Ashok Seth, Farzana, Dr. Farzana, Vice Chancellor of Hera University, and eminent person sitting before me, and very eminent and respected audience. I don't know whether I should thank Abbas, Dr. Professor Abbas Rizvi for inviting me, or I should protest. Because he has invited a person who has nothing to do with medicine. I think I am the only person in this row who does not know ABC of medicine. And he has invited a person like me to say something on biomedicine. When I don't know anything about medicine, how can I say about biomedicine? It's there is a couplet in Urdu, बात करनी कभी मुश्किल मुझे ऐसी तो न थी. I feel that I am not in a position to say something about medicine, but I can say a lot of things. A person like Professor Mansoor Hassan is here. You know all Dr. Mansoor Hassan Padam Shri, and about whom I can say that there is a couplet in Urdu, जिनसे मिलकर जिंदगी से इश्क हो जाए वो लोग, जिनसे मिलकर जिंदगी से इश्क हो जाए वो लोग आपने शायद न देखे हों मगर ऐसे भी हैं. Like Mansoor Hassan, we have amongst us. Professor Abbas Mehdi, right from the very beginning, is a dedicated and devoted young man, and he is doing a lot of things for the benefit of mankind. I am very happy to see that he has been instrumental in getting this organization established in Lucknow. When I received an invitation from Dr. Abbas, I don't know what to say. Then I requested some of my friends to send something about biomedicine. And I got this note, which I'm going to read. I have not written it. I'm simply reading it. In order to promote translational research, Indian Academy of Biomedical Sciences, IABS, was established in 2012 with the vision, keen interest, and efforts of various scientists from India and abroad. Initially, the initiative to form the academy was taken by Professor Hari Sharma from the Netherlands. Now, Professor Hari Sharma is here, and we are really very proud and privileged to see him face to face that he is here. Congratulations, Professor Sharma. Professor Sharma discussed this idea with Professor Abbas Ali Mehdi during one of his visits to his laboratory at King George Medical University. Thereafter, Professor Mehdi got the academy registered at Lucknow on 7th February 2012. Thank you very much, Professor Abbas, for getting it registered here in Lucknow, so that we are greatly benefited by your initiatives. The main objective of the academy was to disseminate knowledge and academic excellence in the field of biomedical sciences. The labs organizes CME workshops, conferences with the primary aim to foster collaboration between various laboratories and scientists to promote biomedical research and innovations and academic excellence in the field of biomedical sciences. Major thrust of academy is to take research from the laboratory to clinics, bench side to bedside. Well, bench side to bedside. I cannot fully understand the whole thing, but since I have received it, I am reading. As I told you, This I have not written anything about it. And the vice versa from bedside to bedside. Biomedical science is the science devoted to the study of the processes of life, prevention and treatment of diseases, and genetic and environmental factors related to health and disease. This branch of science includes various aspects of both the physical and life sciences and uses molecular biology and biotechnology techniques to study diseases and biological processes with the objective of developing effective cures and treatments. According to the WHO, biomedical research has dramatically increased life expectancy. For instance, the life expectancy in America in 1900 averaged 49 years, while this figure had grown to about 69 years by 2004. Now it must have been something more. Moreover, a study established in Lancet showed that in India also, life expectancy has risen from 59.6 years in 1990 to 70.8 years. Wonderful. In 2019, ranging from 77.3 years in Kerala to 66.9 years in Uttar Pradesh. This is very happy news. I must congratulate you. So this is what I can say about biomedicine, because as I told you, I have not written this paper. Some friend of mine has sent it to me. Now I conclude with a remark that first of all, I welcome all those guests who have come from abroad, who have come from out of Lucknow. Lucknow is a place, a center of culture, a center of civilization, a center where you have poetry, you have everything, whatever, the finest traits of life can be seen and can be judged, can be tasted, and can be experienced in Lucknow. A person, Dr. Johnson once said, a person who's tired of life, a person who is tired of London, he's tired of life. Now, Mr. President, I can say that if a person who is tired of Lucknow, he's tired of life. Lucknow gives you everything, whatever you need. And I can tell you that throughout the world, wherever you go, you will not be able to find a city like Lucknow, people who Lucknowites, people like Lucknow, anywhere in the world. Professor Sabra Habib is here. She has traveled so much in the communist countries, in Soviet Union, but she must have not found a city like Lucknow. Am I right or not? Yes. So, Professor Abbas Mehdi, I congratulate you. Professor Farzana Mehdi, I congratulate you. You are heading an organization which has done a very memorable, commendable, appreciable work during Corona period. We can never forget those dark days, and we can never forget the way you and the Chancellor of this institution, Mohsen Ali Khan, has rendered memorable services, always serving the people, serving patients. Sometime I rang him at 2 o'clock, some patient, but Mohsen was always there to help those. I must say that such people are very rare. People are everywhere. There's no dearth of human beings. As Coleridge has said, water, water everywhere, not a drop to drink. So you have men everywhere. लेकिन क्या होता है, कोई इंसान नजर नहीं आता, हो गई कता नस्ल आदम क्या. It is very difficult to find a human being. And बस के दुश्वार है हर काम का आसा होना, आदमी को भी मयस्सर नहीं इंसान होना. So it is very difficult to be a man, to be a human being. And this university, Era University, under the dynamic leadership of our young Chancellor, young Vice Chancellor, and former Vice Chancellor, Professor Mehdi, his able colleague, his professors, doctors, staff, you have made us proud. Really, you have made us proud. I congratulate you and I wish you all success. I hope that you must kindly excuse me that I was not able to say anything about biomedicine. But this is not mine. नकल किया है, मैंने नहीं कहा है ये. This is not mine. Someone gave it to me. I have read it only. So otherwise, if I would have said something of biomedicine, तो लोग यही कहते बक रहा हूँ जुनून में जाने क्या क्या कुछ न समझे खुदा करे कोई. So thank you very much. Once again, I thank you, Professor Mandy, for inviting me and giving me an opportunity to meet such eminent persons, such persons who have devoted and dedicated persons. I must thank you. I must, Farzana Sahaba, I must thank you. And I'm very happy to meet Professor Churamani. It was a pleasant surprise to see Professor Churamani is here, former Vice Chancellor of King George Medical University. She has been very popular. While she was in Lucknow, everybody, there was no restriction in meeting the Vice Chancellor. She has come to Lucknow again, and you will keep on coming, because once a person comes to Lucknow, he keeps on coming to Lucknow. Thank you very much, gentlemen.
Thank you, sir. It is my honor to invite our guest of honor, Padma Shri Dr. Mansoor Hassan, to address the audience.
हाँ वो बैठे तो हैं, देख रहे हैं यूनिवर्सिटी गेट को, एंट्रेंस को देख रहे हैं, हम लोगों की तरफ देख रहे हैं. ठीक है.
Professor Churamani sir, the distinguished guests, Dr. Amar Saab, Sheikh Saab, Akur Saab, Lieutenant General Puri is not here, Farzana, and of course Abbas Saab. मैं आपके सामने बजाय अक्ल की बात करने के दिल की बात करूंगा. और दिल की बात यह है कि it is amazing to see this institution. I saw it when it was just being thought of, and I'm delighted to see authenticity. One of the rarest things in this life now is authenticity. There are people who will talk well. There are people who will make brilliant statements, written brilliant writings, writing up the Constitution, giving speeches of outstanding wisdom. But to create an institution like this in our province is really a highly creditable and unbelievable. मैं दिल से आप सब से कहता हूँ कि let us give a hand to Professor Mehndi and his colleagues. उन्होंने मोहब्बत का एक निशान पैदा कर दिया. This is not just an institution. It is a dream which has come true. लखनऊ में भी अलावा KGMC ये सब पुराने कॉलेजेस हैं, इनमें अपनी जगह है, लेकिन इस तरह का एक कॉलेज क्रिएट करना, इस सिंसियरिटी से क्रिएट करना, और अब और बना रहे हैं, कितनी बड़ी बात है ये, कितनी खुशी की बात है. What an honor, what a joy, what a... सुनता हूँ बड़े गौर से अफसान है हस्ती, कुछ असल है, कुछ ख्वाब है, कुछ तर्ज अदा है. किस खूबसूरती से आपने, किस ह्यूमिलिटी से आपने इस काम को किया है. That is what inspires me. That is what really touches me. और मैं अपनी बात लंबी ज्यादा नहीं कहूँगा, क्योंकि बातें तो बहुत हो सकती हैं. मैं इस वक्त दिल से, दिल की गीता के कुछ अल्फाज आपके सामने पेश करना चाहता हूँ. माफ कीजिएगा, आपको लगेगा कि ये कोई, आप यकीन कीजिए कि बचपन से मैंने 5 वक्ता नमाज सीखी, मैंने हज किया, मेरी वालिदा ने 3 बार हज किया, और बचपन में मुझको संस्कृत और गीता पढ़ाई गई. पंडित सुंदरलाल ने मुझे, जो गांधी जी के साथ थे, मैं 85 साल का हूँ, मैं गवाही देता हूँ अल्लाह के सामने कि मैं जो कुछ भी हूँ इन बुजुर्गों की मोहब्बत जाहिर है अल्लाह की तरफ से. और क्या सिखाया है इन्होंने. सुन अर्जुन मैं हूँ आत्मा बिल यकीन, जो है जानदारों के दिल में मकीन. मैं हूँ मिस्ल जां अहले जां के अंदर छुपा, मैं अव्वल में आखिर में दर में. है आदित्यों में मेरा विष्णु खिताब, मैं अशियाए पुरनूर में आफताब, मरीची मुर्रवतों के अंदर हूं मैं, मनाजिल की तारों में चंद्र हूं। समझ मुझको वेदों में तू वेद साम ऐ होशमंद, तो छंदों में गायत्री का हूं मैं छंद। महीनों में मुझको अघन कर शुमार, बहारों के फूलों में हूं मैं बहार। मैं रूवरों के अंदर हूँ शंकर दलेर, जो है राक्षस यक्ष उनमें कुबेर। तो वासुओं में अग्नि मुझे तू समझ, तो ऊंचे पहाड़ों में मेरू समझ। जो पुरोहित हैं इनमें बृहस्पत हूं मैं, सुन अर्जुन सर करदा पुरोहित हूं मैं। शिकन दहले लश्कर के अंदर कहो, तो झीलों के अंदर समंदर कहो। भार्गव यानी ऋषियों का सरदार हूं मैं, सुखन में सुखन हर पे ओंकार हूं मैं। यज्ञों में हूँ यज्ञ जप निराला हूं मैं, जो मोहकिम है इनमें हिमाला हूं मैं हूँ देवताओं में प्रहलाद सुन, मैं वक्त उनमें जो रखें गिनती का गुण। मैं शेरे बबर सब दरिंदों में हूँ, मैं विष्णु का शाहीन परिंदों में हूँ। मैं सरसर हूँ उनमें जो है तेज काम, मैं तेग और शमशीर वालों में राम। मछलियों में मगर मुझे जान तू, तो नहरों में गंगा मुझे मान तू। मैं आपके सामने हिंदुस्तान की सिंथेसिस, इस ब्यूटीफुल सिंथेसिस, this beautiful coming together, the ancient wisdom of India and the later spiritual entry into it of other religions. It's a unique country. It is a unique place. Let us feel not proud, but grateful to Allah that He has given us this opportunity to work and do something for our country. Let us appreciate those people who are sitting there, their sincerity, their authenticity, and your patience and love and affection. अल्लाह ताला आपको सबको कामयाब रखे और आप सब लोगों की जिंदगी कामयाब हो। मैं सिर्फ इतना कहना चाहता हूँ और खत्म करता हूँ। दिल में किसी के राह किए जा रहा हूँ मैं, कितना हसीन गुनाह किए जा रहा हूँ मैं। ऐसी भी एक निगाह किए जा रहा हूँ मैं, जर्रों को महरूमा किए जा रहा हूँ मैं। आगे कदम बढ़ाएं जिन्हें सूझता नहीं, रोशन चिराग राह किए जा रहा हूँ मैं। गुलशन परस्त हूँ, गुल ही नहीं मुझे अजीज, कांटों से भी निबाह किए जा रहा हूँ। मैं आप सबको मुबारकबाद देता हूँ और इंस्टीट्यूशन के लोगों को, प्रोफेसर अली मेहंदी और उनकी बेगम साहब, वाकई आपने दिखा दिया कि सिंसेरिटी और ऑनेस्टी और इंटेग्रिटी क्या चीज है। अल्लाह परमात्मा, अल्लाह आपको मुबारक करे और आपके लिए आने वाली जिंदगी आपका जो ये सॉलिड कंट्रीब्यूशन है इंसानियत के लिए, ये किसी स्पेशल कम्युनिटी की बात नहीं है, ये इंसानियत के लिए। यू हैव स्टूड फॉर ह्यूमनिज्म, फॉर इंसानियत एंड फॉर एक्सीलेंस इन ह्यूमनिज्म विदाउट एनी ईगो। मैं रसूलुल्लाह के इस लफ्ज के बाद बात को खत्म करता हूँ। किसी ने रसूल करीम से पूछा कि जन्नत में कौन जाएगा, फिर वही आती है, ए रसूल तुम लोगों से कहो कि जन्नत में वो आएगा जो अपनी अना को खत्म कर, जो अपनी मैं को खत्म करते, who annihilates their ego in the love of God। मैं क्या कह सकता हूँ इसके बाद, मैं सिर्फ ये कह सकता हूँ कि अल्लाह मुझे हिम्मत दे कि मैं अपनी ईगो को और भी खत्म कर दूं और कुछ न रह जाए सिवाय अल्लाह के नूर के। उस नूर ही नूर सर बसर सल्ले अला मोहम्मद। हुस्न की दो मिसाल क्या, पहुंचे वहां ख्याल क्या, नूर ही नूर सर बसर सल्ले अला मोहम्मद। मैं इस बात के बाद कुछ नहीं कहना चाहता और मैं सिर्फ ये कहना चाहता हूँ कि आप लोग इस नाचीज 85 साल के बुढ़ऊ के लिए दुआ फरमाएं कि आखिरी वक्त तक वो अल्लाह के और इस प्यारे मुल्क की मोहब्बत में
ये कटेंगे देख तो रहे हो टीवी में ना, तुम बोलने वाला तो जरूरी है।
Thank you, sir. I would like to invite Dr. Abbas Ali Mehdi to announce the names of awardees and fellowship recipients.
अम्मार साहब कह रहे थे कि यहाँ पे वन फिट है, he is the most fit person because he is a teacher. Basically politician तो बाद में बन गए, ये तो टीचर थे, पहले शेख कॉलेज में पढ़ाते थे और प्रिंसिपल रहे वहाँ पे। तो आप अपने को मिसफिट ना समझिए, बायोमेडिकल कुछ नहीं है, जिंदगी क्या है अनासिर में ज़हूर तरतीब, और मौत के आए नहीं अज्जा का परेशान होना। तो सारी जिंदगी और मौत जो है वो अनासिर पे है, और आपने शुरू किया है डेवलपमेंट, तो टीचिंग से ही सब कुछ होता है। तो आपने जो टीच किया लोगों को, उसकी वजह से आप यहाँ पे हैं, पॉलिटिशियन होने की वजह से नहीं है आप यहाँ पे। It's a proud privilege to announce the awards for 2020 and 2021 and 2020 and 2021. Its first is Padma Shri Dr. Mehdi Hassan Award, and we are proud that this has been given this year to one of the favorite students for whom Dr. Mehdi Hassan, I know, being his son, was proud of, is Dr. Ashok Seth, who is the chairman of IABSCON Heart Institute. I'd like to request that the honors may be brought, and I request Dr. Ashok Seth to come forward along with Dr. Venkatesh, Dr. Hari Sharma, Dr. Mansoor Hassan, and Dr. Ammar Rizvi to give the honors to Dr. Ashok Seth. Again, I request Dr. Venkatesh to give the medalling to Dr. Ashok Seth. And I request Dr. Hari Sharma to give the Angvas to Dr. Ashok Seth. And I request Dr. Mansoor Hassan to give one certificate. Raghu sir. I request Dr. Ammar Rizvi Sahab to give the certificate and the other certificate of the award by Dr. Mansoor Hassan. The fellowship certificate by Dr. Mansoor Hassan. Dr. Raghu and Dr. Mansoor Hassan, please. Dr. Ashok Seth is the holder of Padma Shri, Padma Bhushan, BC Rai National Award. He has been awarded doctorate from 6 national universities, including Aligarh Muslim University, his alma mater, Banaras Hindu University, Jamia Millia Islamia, and other universities. He is the founder, advisor of lot many institutions. He has trained hundreds and thousands of cardiologists from around the globe. He has got more than 450 publications, 30 textbooks in cardiology and medicine. He has been acclaimed internationally, being the member and editor of lot many journals in cardiology. He has trained cardiologists from Malaysia, Bangladesh, SAARC countries, China, Nepal, Hong Kong, Indonesia, Afghanistan, Turkey, Libya, and around the globe. So it's our proud privilege that today we are getting honored in honoring Dr. Ashok Seth. Now the next award, Dr. Raghu, you please stay back, and Dr. Hari Sharma. Dr. Seth, abhi ruk jaiye. The first award in the line now is Professor Sohail Ahmed Award for 2021. It goes to Dr. Alpana Sharma from Department of Biochemistry at All India Institute of Medical Sciences, New Delhi, who has already received more than 10 awards. She has published 160 publications and has more than 10,000 citations to her credit. So it's our proud privilege that Dr. Alpana Sharma, my senior from Aligarh Muslim University, I, Dr. Afrozul Haq, who is the Vice Chancellor at Manipur University, he is also sitting over here, and Dr. Alpana Sharma, we all three are the students of Dr. Sohail Ahmed, and it's our proud privilege that today this award has been given to Dr. Alpana Sharma, one of the brightest students of Dr. Sohail Ahmed from Jain Medical College, AMU Aligarh. Dr. Asadullah Khan
Dr. Asad, the award, Dr. Sohail Ahmed Award for 2020 is being given to Dr. Asadullah Khan, again from Aligarh Muslim University. He is from the Department of Biotechnology. He has published over 250 papers. He is a fellow of Royal Society of Chemistry. He has more than 10,000 citations, and he has already bagged 10 awards. So Dr. Asadullah Khan, he bags the award for 2020. Last year, we couldn't have the convocation because of COVID. Dr. Ashok Setabhakir. Next is in line is Dr. Srimati Kusum Sharma Award. It goes to Dr. Pallavi Mitra, who is a DST Inspire faculty at University of Hyderabad. She is a young girl who has been awarded Dr. Kusum Sharma Award, which has been instituted by her daughter, Dr. Belu Sharma from Netherlands, and Dr. Hari Sharma, her son-in-law. And this year, this young girl bags this award. She has already published 16 papers with over 500 citations. So let's clap this young girl from University of Hyderabad, Dr. Pallavi Mitra, who bags the Srimati Kusum Sharma Award for 2021. Next, Shri Om Prakash Sharma Award. And I'm proud to announce that my student, Dr. Kamla Khan Shukla, an associate professor at the Department of Biochemistry, All India Institute of Medical Sciences, Jodhpur, has bagged Shri Om Prakash Sharma Award, which has been instituted by Dr. Alkna Sharma in memory of her late father, Shri Om Prakash Sharma. So it's my privilege that Dr. Kamla Khan Shukla, my PhD student, with 40 papers, with over 2,000 citations, 5 awards, he bags this award for this year. It's for the young scientists in biomedical research. Next is Mrs. Farah Diba Haq Award, and I'm happy that both the awardees are present. First is Dr. Alia Shah. I'm proud that Dr. Alia, an alumni of Aligarh Muslim University, presently working as an assistant professor at Skims Middle College, Srinagar, with 25 papers and lot many citations. Alia bags Mrs. Farah Diba Haq Award, instituted by Dr. Afrozul Haq in memory of her late wife, Mrs. Farah Diba. The award for 2020 is backed by Dr. Amit Kumar from Radiation and Photochemistry Division, BARC, Mumbai. Dr. Amit Kumar has got 103 research articles with over 3,000 citations and 4 awards. This young man bags Mrs. Farah Diba Haq Award for research in cancer biology. Next is Mrs. Abida Mehdi Award for 2020. It has been bagged by Dr. Prasan Priya Naik from Department of Physiology at All India Institute of Medical Sciences, Jodhpur. Dr. Naik has got 50 publications with 1,200 citations and 4 awards. Dr. Naik, if Dr. Sadhna Sharma is here, Dr. Sadhna Sharma from Patna, because Sadhna Sharma too are there. Sadhna Sharma from Chandigarh and Patna. Sadhna ji, please. I'm happy that the award for 2020 has been backed by Dr. Saurabh Vashni, who was supposed to come here. Dr. Vashni is Director, IABS, Deoghar, Jharkhand, and he's also holding the, he is having the charge of Director, IABS, Patna. He has bagged Mrs. Abida Mehdi Award for 2021, and Dr. Sadhna Sharma is a professor and head department of biochemistry at All India Institute of Medical Sciences, Patna. Today, Dr. Saurabh Vashni, he couldn't come because of some important meeting. So he requested me to hand over the award to Dr. Sadhna Sharma. Dr. Sadhna Sharma, please receive the award on behalf of Dr. Saurabh Vashni, Director, AIMS Patna and AIMS Dugar. The next award I'm going to announce, which will be a surprise for the students. This is Professor Nurul Islam Award. And this award this year has been backed by a tough competition this evening only by the Dr. Meenakshi Sharma from AIMS New Delhi. Meenakshi is present here. Dr. Meenakshi. Dr. Meenakshi. She's here. Meenakshi, please come forward. It's a surprise for her because she must not be knowing it. This is being announced just here. It's a tough competition between 7 and she's just got to know it. So Professor Nurul Islam Award instituted by Dr. Najmul Islam in memory of his late father, Dr. Nurul Islam, is backed by Dr. Meenakshi Sharma from AIMS New Delhi. Congratulations, Meenakshi. Mr. Santosh Raizada, please come forward. Next award I'm humbled to announce is Lifetime Achievement Award, which is given for 2020 to our dear friend, Dr. Professor Arun Raizada, who we lost in COVID. He was president of IABS. He hosted the meeting of IABS at Delhi with great fanfare. And today we are having his son. Dr. Seth must be knowing Dr. Arun Raizada. He was our former president. So I request Saurabh to come forward and receive the award on behalf of his late father, respected Dr. Arun Raizada, who is not with us now today, but his memory, his work, his affection for all of us remains here. And it's our proud privilege that his son, who's carrying his legacy, is here. So I request Dr. Seth and Dr. It's an emotional moment for him. I can understand. So I request Dr. Hari Sharma and Dr. Ashok Seth to do the honors. Dr. Raizada worked with Dr. Ashok Seth at Sports. So that's why Dr. Seth has got memories of him. Beta khush raho. All the best, beta. Now we are going to announce the fellowships. In the list, first is Dr. Professor Afrozul Haq, Vice Chancellor at Imphal University, Manipur. I request Professor Afrozul Haq to come forward and receive his fellowship. Dr. Raghu and Dr. Hari Sharma, please give the honors. Next is Dr. Afrozul Haq have got 85 papers published, and he's an international authority on vitamin D because he have carried out much work on vitamin D. Next is Professor Ajay Singh. It's my proud privilege to announce the name of Dr. Ajay Singh, Director, PGI, Child Health at Noida. Dr. Ajay Singh has got 180 papers to his credit with more than 2,000 citations. And Dr. Ajay Singh is doing tremendous job. He established the Department of Pediatric Orthopedics, where we are going to start shortly MCS at King George's Medical University. And thereafter he moved at PGI CH Noida, which is he is heading at the moment. Congratulations, Dr. Ajay Singh. And we are proud of our association. Thank you. Next is Professor Ashutosh Haldar from Department of Reproductive Biology, AIMS New Delhi. Dr. Haldar has got 83 papers to his credit with 1,500 citations and many awards to his credit. Next is Dr. B.S. Shankanarayan Rao from NIMHANS, Bengaluru. Another great friend of IABS, great friend of ours, who always comes and starts the meetings of IABS from the beginning till end. So this is his contribution to IABS. He's got more than 150 publications in high impact journals like Neuron, PNS, Journal of Neuroscience, with 5 books, 58 book chapters, 10,000 citations, 22 awards. We are proud of Dr. Rao. It's our proud privilege that we are feeling honored by giving the fellowship to Dr. B.S. Shankaran Rao from NIMHANS, the prestigious institution on neurosciences at Bengaluru. He is currently the registrar of the institution also. Next is Professor Farzana Mehdi, who is heading this university at the moment. She has got 75 papers with over 2,000 citations and many awards to her credit. Congratulations, Dr. Farzana. Next is Dr. Jyotideep Kaur from Department of Biochemistry, PGI Chandigarh. Dr. Kaur is another friend of IABS. She has been coming to all meetings and she is doing tremendous work at PGI Chandigarh. Today we heard her talk today in the morning. She has got 90 papers to her credit with 2,500 citations. Congratulations, Dr. Kaur. I hope that our association will continue and you will continue to patronize IABS. Next is in line is our president, Dr. KG Raghu, who is an eminent scientist from CSIR, friends of ours. He has the distinction of being president of IABS twice. He has got 90 papers with 2,700 citations, many awards, and he hosted a wonderful meeting at Trivandrum few years back. So we are proud. Dr. Churamani also attended that meeting. I think Madam remembers it. Next in line is Professor Najmul Islam from Aligarh Muslim University, Jain Medical College. He headed the Department of Biochemistry there. He's Professor of Biochemistry, my childhood friend. He has got 90 papers, more than 3,000 citations, many awards to his credit. Dr. Najmul Islam. He has been the president of IABS. He hosted a meeting of IABS at Aligarh, and we are proud that he is the proud recipient of IABS fellowship. Next in line is Professor Ramesh Chandra. He needs no introduction. He has been the ex-vice chancellor, the builder of Bundelkhand University, Jhansi. He has been the director of Ambedkar Institute at Delhi University. He is the director of nanotechnology at Delhi. He is a fellow of Royal Society of Chemistry, London. He is an outstanding researcher in biomedical sciences. He started the Department of Biomedical Sciences at Jhansi, I remember. He is our friend. He has got the credit to be amongst the top 2% scientists ranked by the international agencies. Dr. Chandra, we are proud to have you. Next in line is Professor Sadhna Sharma, again from Department of Biochemistry. She is heading the Department of Biochemistry at PGI Chandigarh. Dr. Sadhna Sharma, she has got 90 papers with over 5,000 citations, many awards to her credit, and she's doing tremendous job as a head Department of Biochemistry, I know, at PGI Chandigarh. She is carrying that legacy of stalwarts like Dr. Nath at Chandigarh and Dr. Rajan Prasad, our friend. So, Dr. Sadhna Sharma, we are proud to have you as a fellow of IABS. Congratulations. Next is Professor Suman Kundu, Director BITS Planning, Goa Campus. Dr. Kundu? Yeah. So thank you, sir. Thank you very much. I think with this, we finish with the awards and fellowships. Thank you.
ठीक है पूरा स्ट्रेट पूरा उधर ले जाओ। नहीं क्या कर रहे हो? अबे उधर ले जाओ, उल्टा कर रहे हो। मेरी तरफ लाओ। हाँ और आओ
Thank you, sir. It is my honor to request Padma Bhushan Dr. Ashok Seth to address the gathering.
Eminent dignitaries on the dais, truly some of them very eminent. The Honorable Vice Chancellor of IRA University and the trustees of IRA University. Dear friends, scientists, doctors, students in the audience, and a number of friends. Let me also, by name, address Professor Mansoor Hassan Saab. The reason for that specifically is that I wouldn't be here if it wasn't for the blessings of teachers like Mansoor Hassan Saab. And that he knows it, that when we returned from abroad 30 years ago, 33 years ago, to create the Escorts Heart Institute, and he was and still is the dawn of cardiology for India. He actually had his blessings clearly defined for me with a lot of affection, and I have a huge amount of respect. When he called me by the first name, even 30 years ago, and said, "Jab aaye ho wapas, to Hindustan ke liye aapne kuch karke hi dikhana hai, isko wapas dena hai." And he said that the blessings are with you because Escorts Heart Institute got created by a number of alumni of the KGMC. And some of them were his students who then became my students. And he kept passing on his students to me to train further. And all of them have become luminaries. So, aapka jo aashirwad, aapki jo duaएं हमारे साथ हमेशा हैं, इसलिए I have a huge respect for him. And then, of course, I must say it's a privilege and an honor to be given Professor Mehdi Hasan Saab Award. Those of you who did not know him or never had the fortune of being closely associated with him or being taught by him have missed an opportunity in your lives of one of the greatest teachers who ever existed. And that's why I'm humbled when I receive his award, because we entered medical college, JNMC. His fame and name and aura was far and wide, beyond the medical colleges, beyond the walls of AMU. It was national, that when you hear him teach, when you learn from him, you are already halfway to succeeding. And that was true, phenomenal. If I was to tell you the few things I remember about him, and it is just right for me to say that, because many of you don't know, and many of you may never have met him. He was not just a professor and head of the Department of Anatomy. This was one of the most talented individual doctors you could find in the country, who grew up in very difficult circumstances, with his parents having passed away when I think he was very early childhood. And yet, with the help of his uncles and aunts, carved out an exceptional career, not of a doctor, but of a teacher. He would come to the auditorium, the college auditorium. In that 1.5 hour of a lecture of anatomy, he would mesmerize you, because he could draw the atlas of anatomy in color with chalk to create images which were so well understood that you could photograph them and it would be better than the album of anatomy that you actually had printed with. It was amazing. He drew with 2 hands, and he would draw the muscles, and he'd draw the tendons, and he would draw them in different colors, picking up chalks and throwing chalks. And as he turned around, you would have an art of anatomy. But that was not just it. He was such a profound teacher that he applied that anatomy to every discipline of medicine in a way that when we qualified and did our first professional and moved on to the second professional and then went to the third professional and had exams of surgery, medicine, ophthalmology, you name it, he had applied anatomy to them. So it was our every student would go up to him, every batch would go up to him and say, during the third professional exam, would you teach us surgery? Would you teach us applied surgery? Would you teach us applied ophthalmology? Would you teach us applied obs and gynae? And he would translate his knowledge of anatomy to actually what was, and he would take this time out, by the way, this was not his job. He would take from 6 to 9 continuous classes for us, even in the final year. That was education, that was a teacher, that was a phenomenal teacher. And therefore, I think there is not one student of JNMC who cannot, apart from just pray for him, touch his feet, respect him, because we've never seen a phenomenal individual, and India is probably never going to see such a phenomenal teacher ever again, who actually had into his heart, teach, educate, smile. And by the way, that was not just a teacher, he was an all-rounder. He actually created the first brain research laboratory in India after his 2 or 3 year sabbatical in Germany. First brain research laboratory. He was a scientist, he was a researcher, he was a neuroscientist, he was a player, he was an avid cricketer. He was always the the college teacher in charge of all our activities. He was a dramatist. He actually was a footballer. I think he was many people rolled into one. And then when I see this, and I see this, one of his visions, which have translated into this creation of Era Medical College and Era University. And I remember the time that he actually put down his efforts into creating this. I think that we are very, very privileged and proud to stand in his footsteps. I would say in his footsteps and enjoy this lovely evening. And this is phenomenal, phenomenal. Now, I don't want to talk too much here. I mean, you're already probably would be saying, "Bahut ho gaya yaar, khana khaye." But I just come, I think, you know, when I stand at the Era Medical College, there's one thing that I have to say. And I was asked very recently in a what they call the greatest of all time interview. And the greatest of all time meant कि जी 2-3 ऐसे doctors हैं, interventional cardiologists हैं, जो कि greatest of all times हैं, and I just happened to latch into one of those positions. And I was asked this question in a national interview saying, how do you become the greatest interventional cardiologist? How did you become the greatest interventional cardiologist? And I thought to myself, that's a very odd question. You know, you can say that for cricket players, you can say that for tennis players. कि जब 21 ग्रैंड स्लैम जीते हैं तो आप तो ग्रेटेस्ट ऑफ ऑल टाइम हो गए, और आपने 20 जीता तो उतने ग्रेट तो नहीं हुए, और अगर आपके 18 ग्रैंड स्लैम्स थे तो थोड़े कम ग्रेट ऑफ ऑल टाइम्स हुए. But how can a doctor be great? So I said, and I want to pass this thought process on to you, कि डॉक्टर तो गुड ही हो सकता है, ग्रेट तो कभी नहीं हो सकता. It is not supposed to be a great doctor. Let me explain to you why I say that. देखो, the greatness is something which is a perception. Good हो सकते हैं because there are we are privileged to be in a profession where we are blessed, where we bless every day by thousands and thousands of people. उनकी दुआएं हमारे साथ रहती हैं, and we are only doing a job. कोई ऐसी job नहीं है, कोई ऐसा profession नहीं है where you receive blessings of thousands and thousands of people every day just because you're doing what you're supposed to do. Now the greatness is, there is only 4 things that you want to do. And before being a great doctor, you have to be, before being a good doctor, you've got to be a good person. and a good human being and a good man. So I always start off by saying, you're a great interventional cardiologist. I say, great, nahi, good interventional cardiologist, or uske liye you got to be a good cardiologist. And if you got to be a good cardiologist, you got to be a good physician. And if you got to be a good physician, you got to be a good doctor. And if you got to be a good doctor, you got to be a good human being. And that's the full stop. I would not want to be remembered as the greatest interventional cardiologist India had. I wouldn't be wanted to remember as the only cardiologist who got honored by 6 national universities with a doctorate of science. I would rather not be known as the most skilled cardiologist. I would never want to be known as the guy who earned a reasonable amount of money or who drove something or didn't drive something. I wish that when I die, people just remember me, that he was a good doctor. And more importantly, he was a good human being. And for that, I always say, I just need my 4 C's, which I still practice and believe in. And that is compassion. That's the first thing we as doctors need. With our patient, understanding him, empathizing with him, caring. The second C, and if we say the second C is caring in the 4 C's. So we have compassion and we have care. It should never strike you to leave a patient unwell, even if it means staying there for 24 hours. You go walk out of your house saying, God, please do everything to save my patients. You come back with successes of the worst cases that you've treated, and you brought the patient out and you say at home, just 3 words that you've got to say in front of God. Thank you for keeping my patient alive. After all, he was somebody's father. He was somebody's brother. That lady was somebody's mother or somebody's wife or somebody's sister. And if you do that, that is care. It should hurt you if a complication occurred. It should give you sleepless nights, and you should be able to spend that time. The care revolves around that agar aapke haath se complication ho bhi gayi, aapke haathon se to nahi hoti hai. If you take full precautions and care, it is inevitable that some people will have a complication which was unavoidable. But aapki care is level ki honi chahiye. Even if the patient doesn't survive after 10 days of your trying your best, if a month later the same patient's relative bring you another relative of theirs and say, Doc Saab, iski treatment to aap hi ne karni hai, iski angioplasty to aap hi ne karni hai, then you know that you did your best, you gave your best, irrespective of outcomes. Then we have competence. I mean, we can't just be cowboys. I have pioneered, and I think everybody knows, innumerable techniques into this country for the first time in Asia, for the first time in India, path-breaking technologies and techniques. I was asked the same question also. What takes you that courage to actually do that? And you always say, it's not a courage. India deserves the best, and India deserves to actually have the best of science, best of technologies, as you're giving, which the world should recognize. And our Indians deserve the best in every sphere. And if I believe that God has willed that I would give it to them, then so be it. I go in with the confidence of God's what he's got to do. But at the end, we can't be cowboys, and we've got to upgrade ourselves. We've got to learn. We've got to actually keep on upgrading our skills and education and evidence-based medicine practices. So that's my 3rd C: compassion, care, and competence. And of course, finally, I would say conscience. C stands for the biggest C in my life, is conscience, ethics, evidence-based practices. आप सो ही नहीं सकते अगर आपने ethical medicine नहीं practice की. In the changing circumstances of the attraction of lucre, should we say, we see a deterioration of ethical practices in all spheres of profession. And that is also infiltrating into our medical profession. I say it for the students, I say it for the young gen, that आप कैसे सो सकते हो अगर आपका वो पैसा है जिसके बेसिस पे आप अपने बच्चों को घुमाते हो और अपने स्पाउसेस के लिए कोई प्रेजेंट लाते हो, और आपको ही पता है वो पैसा सही नहीं है, वो पैसा is marred with some unethical practices. कैसे आप किसी को फेस कर सकते हो? आप अपने बच्चों को कैसे फेस कर सकते हो? आप अपने स्पाउसेस को कैसे फेस कर सकते हो? आप माँ बाप को कैसे फेस कर सकते हो? It is absolutely unthinkable that conscience should hurt us. The moment we do something wrong, look at the patient who sits in front of us and think of that as your own brother, your own sister, your mother, your father. Give to that patient what you give to yourself. I say, agar we have lots of ambiguities in medicine, there's huge amount of gray area, which you can't decipher, but don't use that gray areas for your own conveniences on that particular patient in front of you. अगर वो gray areas आपने define किए हैं अपने लिए कि मैं अगर मेरी ये disease होती तो मैं stent डलवाता और मैं कौन सा stent डलवाता या मैं operation करवाता तो क्या करवाता. If that is your criteria, put it to that patient in front of you. Otherwise, he's your relative. You will give him what you give to your own relative and friends. So I think, do I have 2 minutes more? So that it encompasses that thing about the greatest doctor. There is nothing like greatest. But then he asked me, how did you become so successful? What are the mantras for success? And I said, ये भी एक बड़ा अच्छा question है. What are the mantras for success? And I said, I think I'm not successful. I think you are perceiving that I'm successful. So the mantra for success is your perception, not that I became successful. Because success is an arbitrary definition in different times of your life. For example, when I started my career, I say, I'm going to be the best intervention cardiologist in my expertise. Let me train here, let me train there, let me do new techniques. So मेरा तो वो success था कि मैं सबसे अच्छा expertise दिखा रहा हूँ. 10 years later, I said, यार थोड़े पैसे भी कमाने चाहिए. My success will revolve around अगर थोड़े पैसे आए, थोड़ा घर बड़ा अच्छा हो, मेरे बच्चे अच्छे स्कूल में जाएं, and my success moved on. And then it comes कि success में needs वाली बातों में next 10 years or 15 years wants वाले में चले गई. यार अब तो आर्ट भी खरीदना चाहिए, अब तो ये भी खरीदना चाहिए, कार तो छोटी होगी, बड़ी कार खरीदनी चाहिए. And that success, definition of success goes from needs to wants. And then you come to this age in the mid 60s and 70s, then you say success तो वो था कि अगर मैं family के साथ time बिता सकता, बच्चों को बड़े होते देख सकता, उनकी education पे जरा ध्यान देता, उनकी success पे ध्यान देता. So every day you can start feeling that your whole life was unsuccessful, whereby others perceive that you're successful. So there is nothing like success, but there are 4, if you want to ask me, there are 4 paths to success. And the 4 paths are, there's no question that you have to be ethical, compassion, care that I've just told you. And the second aspect of success is that you just have to have the blessings of your peers, your patients, of everybody that you meet. जैसे मैंने कहा, मंसूर हसन साहब के blessings थे हमारे ऊपर, उसकी वजह से हम successful बने. Your perception of success, at least उसके हिसाब से बने. And then, by the way, let's not forget that there's my family who must have actually sacrificed, usually my parents, my wife, my children who never saw me for days because I kept working till 2 and 3 and 4 o'clock at night. कि अगर मैं 3 दिन travel पे भी चला जाता था तो घर में वापस आता था, उनके लिए कुछ लाता तो कहते थे आप कहीं गए हुए थे, हमें तो पता ही नहीं कि आप गए हुए थे क्योंकि वैसे ही हम आपको नहीं देखते, तो आप बता रहे हो कि आप सिंगापुर 3 दिन के लिए गए थे. So they sacrificed, my wife sacrificed much of a career for my success. So there are people who actually give up and sacrifice their lives so that I could be successful, so that you could be successful. And then finally, never forget the will of God. The God spirituality has to be a part of your success. The success is not mine. I could walk down the steps, fall, bang my head. I would be a vegetable. My driver could bang my hand in the door of the car and I would never do an angioplasty again. वो success जो है ना, 1 minute में जो आपका perception of success है, it takes a split second for it to disappear. और आप एक vegetable हो, आप unconscious हो, आप coma में हो, आपको उसके बाद कोई और पूछेगा नहीं. Dr. Seth doesn't exist anymore. He's gone. So What I want to tell you is, he wills it every day we walk, and you perceive me to be successful, and I perceive you to be successful. Just say, thank God, we're doing our job, we're doing it well, we're giving it our best, and that's what makes it successful. Thank you very much.
Thank you, sir. I would like to invite Dr. Sabra Habib, ma'am, to announce the release of the book.
मतलब वो वीडियो बस 1 मिनट बोलेंगी, 30 सेकंड फौरन वीडियो प्ले करनी, ऑडियो पे स्टॉप फॉलो।
इबादतों की तरह अपना काम करती हूँ, इबादतों की तरह अपना काम करती हूँ, मेरा उसूल है पहले सलाम करती हूँ। For the first time I am nervous. Dr. Ammar Rizvi, Dr. Mansoor Hassan will certify that I have translated prime ministers, chief ministers, governors, ambassadors, and I was never nervous. Why? Today, the galaxy of such prominent persons, rare persons, and remembering Dr. Mehdi, who was very kind to me. I'm sorry, Dr. Abbas Mehdi warned me that not more than 2 minutes. Okay, 1 minute, I have taken one left. Then anyhow, so Dr. Mehdi and I still cashing Dr. Abbas Mehdi that your father was very kind to me and he always helped me. Anyhow, Dr. Ashok Seth, your speech was wonderful, all philosophical. Since Dr. Mansoor Hassan and Dr. Ammar Rizvi ने मेरे अंदर शायरा को जगा दिया है, एक दो कपलेट तो जरूर सुनाऊंगी। डॉक्टर अशोक सेठ, आपके लिए आपने जो कहा कॉन्शियस ज़मीर अंदर देखो, there is a couplet for you. न पीर से पूछो न फकीर से पूछो, न पीर से पूछो न फकीर से पूछो, तुम अपने बारे में अपने ज़मीर से पूछो. So you must ask this thing. ये आपके लिए शेर है, wonderful. I am going to remove the doctor from my name. It will be only Sabra Habib, because such doctors, they are real doctors. Anyhow, thank you very much. We'll talk. Dr. Farzana Mehndi, Dr. Abbas Mehndi के लिए, इस इरा के लिए जिसको मैंने ग्रोइंग होते देखा है, बढ़ते देखा है, and I am a frequent visitor also, कि कहते नहीं है कि जमीन सलाम करे आसमान सलाम करे, जमीन सलाम करे आसमान सलाम करे, कुछ ऐसा काम करो कि जमाना सलाम करे. आज जमाना सलाम कर रहा है इस इरा को. It has a name and they have created it by their compassion, by their humbleness. Dr. Farzana Mehndi, Dr. Abbas Mehndi से ज़्यादा humble और loving और कैरियर तो मतलब एक अनार 100 बीमार है. He is in so much demand that he's hardly seen to shower his kindness. Anyhow, coming to the point, Dr. Amrita Das is here. This is a book, My Green School, written by Thakur S. Podial from Bhutan. This book has been translated into 18 languages. And we are lucky that after English, it has been translated into Urdu. किसे तहजीब कहते हैं वो खुद ही जान जाएंगे, किसे तहजीब कहते हैं वो खुद ही जान जाएंगे, तो अपने बच्चों को फकत उर्दू सिखा. So these things he has, I mean, he has written this book into English. My Green School, totally new idea. And Dr. Farzana Mehndi or Dr. Abbas Mehndi caught and understood the importance of this book, and they supported and they got it published from IRA University. I really thank them and congratulate. Please do read this book. It is into English, written by, forward is by Amrita Das and translated by me in Urdu. He has been ex-minister of education, Bhutan, and My Green School. The book has been translated into Hindi by the IRA University. Little bit about before 2 minutes over. Be kind in the public, please. Professor Podel has been a visiting professor and is a member of several international panels and advisory boards. He remains deeply engaged in public service and reflects on the outer and the inner life of his nation. He has written 3 books, As I Am, So Is My Nation, his book. Second book is Right of Vision and Occasional Views. Third book is The Light of My Life. And I requested Shiraz Rizvi to play because 2 minutes over. Thank you very much for giving me such a august galaxy sitting over here and giving me this chance. Thank you very much. So over to Shiraz, please play. It is Dr. Cordial's message which will be conveyed to you. Thank you very much.
Participants, present to you my warmest greetings and prayers from Bhutan, and thank you for the joy and privilege of being a part of this August gathering. As things stand, this 10th edition of the Indian Academy of Biomedical Sciences International Conference is a testament of faith, an act of courage, and an expression of hope. Over 2 years on, humanity continues to live under the shadow of COVID-19. The invisible enemy is still around, and we haven't seen the last of its variant. We lost millions of the members of our human family across the world. The old certainties no longer remained certainties, and the regular rhythm of our life was thrown off balance. But the good news is that millions more have survived the scourge of the deadly virus and its variants, and our societies are beginning to return to some semblance of the normal. Despite the pain and privations unleashed by the unprecedented challenges, what became absolutely clear to men, women, and children from coast to coast, country to country, continent to continent, is the preciousness of life. Nothing matters more than life, as my most compassionate king so firmly believes in. Yes, nothing is more important than life. And that is the reason we are here. We were caught unawares, but people like you, our scientists, researchers, and medical professionals rose to the challenge and gave to the world life-saving vaccines and vital services that have secured humanity from what could have been total decimation. This conference is a tribute to that spirit of caring and healing in honor of life and living, and that affirmation of the primacy of life and the need to preserve the preciousness of life is, I believe, at the heart of your medical profession. Hello. I commend the bold vision of King George's Medical University and ERA University and the hard work of the organizing committee comprising the departments of biochemistry and biotechnology respectively of your great seeds of learning for making possible this confluence of great medical scholars, practitioners, and policymakers from around the world in your beautiful city of Lucknow, of great awakenings and outstanding achievements. As a privileged beneficiary of the extraordinary breakthroughs in support to life that you and your colleagues across the world have demonstrated, I would like to offer my highest tributes to all of you and your teams, no matter wherever you serve, for your incredible commitment to life and to its preservation. In the course of the next 2 packed days, you will explore recent trends in biomedical research with particular emphasis on current challenges and future prospects. Your insights and experiences will contribute in significant ways to preparing our societies to meet future challenges as you apply your findings to serve humanity ever better at all times. In your infinite generosity, ERA University has considered it worth your while to undertake the publication of the Urdu edition of my little book titled My Green School. I would like to express my deep gratitude to the Honorable Vice Chancellor, Professor Dr. Vision and Initiative. The Urdu edition of My Green School would not have been possible. Above and beyond all is the larger-than-life presence of His Majesty the Fourth King of Bhutan, the Gyalpo Jimmy Singh Wangchuk, from whose all-embracing vision of gross national happiness, my little book derives its vital life and sustaining energy. This conference is a tri-junction of life. The organizing universities have themselves come into being with the primary goal of serving and supporting life through educating in the original promise of the profession. Life, bio, is the defining commitment of the Indian Academy of Biomedical Sciences. And green in my green school is not only the primary color of Mother Nature, but a metaphor for life in all its infinite variety. Therefore, it is hardly a coincidence that all the positive energies have cohered here in this conference in honor of life, precious, various, and beautiful. I'm most grateful to the organizing committee for the gracious decision to launch Shami Amojis at this August gathering. It is my hope and my prayer that the sole message of my little book will bless the speakers of the many languages into which it has been translated. I wish the conference every success and fulfillment and happiness to all of you as you look ahead. Dhanibad, Tashidili, and thank you.
Thank you very much. Thank you. As I told that the book has been translated into 18 languages, and from English, the foreword written by Dr. Amrita Das was translated by me, and the proofreading has been done by me too. Thank you, Dr. Farzana Mehndi, for trusting me. Some corrections have been done. I request Dr. Amrita Das kindly come over to the stage. Please, Dr. Amrita Das, please come.
This is the Urdu version of the book. I request Dr. Seth to kindly release it. Dr. Podial is there in Bhutan. Amrita Das, please come forward. And I'm happy to announce that we started the Department of Happiness at IRA University, and Dr. Podial visited us a few years back, and the Department of Happiness is working in the IRA University for the benefit of all the students, whether they are from medicine, nursing, pharmacy, or allied health sciences. And it is not for just happiness, but it is to overcome stress and distress which medical professionals experience during their stay in hospital settings. So that's what is Department of Happiness here. Good evening, Dr. Podial.
Thank you so much, sir. Thank you so much, sir. We are running short on time, so we have to proceed. Sir, you are muted. You are muted,
Thank
sir.
you, everyone. It is my great pleasure to invite Dr. Saroj Churamani to say a few words.
It's all right. Thank you so much. We'll talk later. The next speaker is on board, sir.
Thank you so much indeed, all of you.
Okay. Thank you, sir. It was a blessing to have you. Thank you so much.
Thank you
Take care.
so much. Thank
इस
you.
Good night, sir.
खुशनसीब
Good night.
शाम में मैं अपना
Good
इसको
night, sir.
सौभाग्य ही मानती हूँ कि आज सब इतने महान लोग इस महान यूनिवर्सिटी में आए हैं और बड़ी महान एकेडमी के फंक्शन में आने का सौभाग्य मिला। ये मेरा खुशनसीबी है। कहा जाता है कि जब आप बड़े महान लोगों के साथ में होते हैं तो ये आपका सौभाग्य होता है और ईश्वर का आशीर्वाद होता है कि आप उनके साथ हैं, क्योंकि अगर आप उनके लायक ना होंगे तो उनके साथ कैसे होंगे। लायक बने हैं तभी ईश्वर का हाथ है। और उससे भी बड़ी बात यह है कि इतने महान लोग हैं। आप सभी जान चुके हैं कि हमारे डायस में जो बैठे हैं कितनी महानतम विभूतियां हैं देश की हैं और जो विदेश में जाकर देश का नाम रोशन कर रहे हैं। इस एकेडमी के जो फाउंडर प्रेसिडेंट डॉक्टर हरीश शर्मा जी अपने देश के हैं, आगरा मथुरा के एरिया के हैं और पूरी एजुकेशन अपने देश में पाई और देश का नाम विदेश में उजागर कर रहे हैं। और इस एकेडमी की संस्थापक वही बने, उन्हीं की प्रेरणा से डॉक्टर मेहदी साहब ने और उनकी पत्नी ने उसको और आगे बढ़ाया है। हमारे बड़े सौभाग्य की बात है। डॉक्टर मेहदी हसन साहब के बारे में, अब्बास मेहदी साहब के पिताजी, मुझे बड़ा सौभाग्य हुआ थोड़ा उनसे परिचय पाने का। ईश्वर की कृपा से कुछ दिन जरूर मैं लखनऊ में आई और यहाँ के KGM की वाइस चांसलर बनी। तब उस समय मैंने देखा था कि यहाँ पे 5 विद्यार्थी ऐसे थे जो एनाटॉमी में पास ही नहीं हो पा रहे थे और उनकी वजह से काफी इंस्टीट्यूशन में एग्जामिनेशन को लेके जब एग्जामिनेशन का टाइम आता था कुछ न कुछ गड़बड़ी बहुत होती थी। और उसकी तह में पता चला वो 5 विद्यार्थी थे जो नहीं पढ़ पा रहे, नहीं पास हो पा रहे। फिर उनके पास नेफेरियस लोगों का गढ़ बन जाता है और फिर उनकी एनर्जी उसमें जाने लग जाती है। तो डॉक्टर मेहदी अब्बास साहब के पास मैं गई और उनसे कहा कि सर इन बच्चों का कुछ कर सकते हैं। बोले हाँ मेरे पास भेज दीजिए, शाम को मैं घर पर पढ़ा दूँ। और बिलीव मी, उन्होंने शायद 15-20 दिन या महीने भर पढ़ाया और वो 5 बच्चे बहुत अच्छे नंबरों से पास हुए। तो ये उनकी कला थी, प्यार था, उनमें टीचर कम और पिता ज्यादा था। तो उन बच्चों को उन्होंने बिल्कुल अपना बच्चा जैसे समझा और इतने अच्छे से पढ़ाया। तो हम बड़े सौभाग्यशाली हैं कि आज हम उनको याद कर पा रहे इस प्लेटफॉर्म से। इसके अलावा मंसूर हसन साहब बनारस लखनऊ के जाने माने कार्डियोलॉजिस्ट तो हैं ही, लेकिन इसके अलावा कितने बड़े अच्छे ह्यूमन बीइंग हैं। उनकी फिलॉसफी जान के आज जब उन्होंने अपना इतिहास बताया, वो बोले कि मैं तो न केवल अच्छा मुसलमान हूँ क्योंकि मैं 5 बार नमाज भी पढ़ता हूँ और वो संस्कार पाए, लेकिन मेरे तो 440-50 साल पहले ये इस देश में जो संस्कार पाए वो पाणिनि वालों से पाए। तो वो अपने आप को कह रहे थे कि मैं उस समय का ब्राह्मण हूँ और आज के समय का तो अच्छा मुसलमान हूँ। देखिए हमें मजहब ये नहीं बताता है कि आपस में कोई भी बैर रखने का। बहुत जरूरी बात है कि धर्म तो कहीं अलग है ही नहीं, और खास करके न तो हिंदू धर्म अलग है ना मुस्लिम धर्म अलग है। हिंदू धर्म की तो व्याख्या उतनी ही बड़ी है जिसमें कि निराकार भी मैं हूँ और आकार भी मैं हूँ। आप जो मुझे श्रद्धा से जो पुकारे वो मैं वही हूँ। भगवान ने कहा आप मुझे श्रद्धा से जो कुछ भी कह दूंगा, जिस रूप में आप कहेंगे उस रूप में मैं आपको दिखाई दूंगा। और अगर आप मानते हैं तो मैं तो सर्वव्यापी हूँ, कण कण में व्यापी है, और यही शायद इस्लाम का भी कहना है। तो हमें आज मैं कहना चाहूंगी कि हम बड़े सौभाग्यशाली रहे कि हमको मंसूर हसन साहब से सुनने उनके विजडम की बातें मालूम पड़ी। यस, हमारे डॉक्टर अशोक सेठ साहब कितनी महान बातें बताई जो जीवन को आगे ले जाने में हेल्पफुल हैं। और हमारे हरीश जी ने, विकेश जी ने, सबने इतने अच्छे अच्छे विचार रखे और ये विचार हम सबको प्रेरणा देते हैं कि हम खुद तो आगे बढ़े ही, पर इस देश को जो सारे जहां से अच्छा हिंदुस्तान हमारा है, इसको और आगे बढ़ाना है और अच्छा बनाना है। और इस देश के जो महामंत्र है पूरी वसुधा ही कुटुंब है, उस कुटुंब के लिए हमें उस आदर्श को आगे बढ़ाते रहना है। हमको मिलजुल के, एक्सीलेंस की रोड अनएंडिंग है और एक्सीलेंस पाने के लिए हमें बहुत ही संभल के रहना पड़ता है, सतर्क रहना पड़ता है, तभी हम आगे बढ़ सकते हैं। तो हम खराब चीजों से अपने आप को हमेशा संभालें, क्योंकि बीमार ही शरीर ही नहीं होता है, समाज भी बीमार होता है। और हम सबका कर्तव्य है मिलजुल के कि हम अपने बीमार समाज को और जल्दी ज्यादा संभालें, अच्छा संभालें, जिससे कि शांति के साथ हम आगे बढ़ सके। जय हिंद, जय भारत।
Thank you, madam. The memories of the magnificent monuments and memorials may slip into oblivion, but a word of appreciation, a word of gratitude may indelibly etch its presence in our minds. With these words, I now request Dr. Hari Sharma sir to kindly do the honor and present a memento to Dr. Ashok Seth. Now I request Dr. Amar Rizvi to kindly do the honor and present memento to Dr. Mansoor Hassan. I request Dr. KG Raghu to kindly hand over the memento to Dr. Amar Rizvi. I request Dr. Venkatesh sir to kindly hand over the memento to Dr. Hari Sharma. I request Dr. Saroj Churamani to kindly do the honor and present memento to Dr. Venkatesh. I request Dr. Farzana Mehdi to present the memento to Dr. KG Raghu sir. Now, I request Dr. KG Raghu sir to please come forward and hand over the memento to Dr. Bipin Tivle sir. Sir, please come. Dr. Bipin Tivle is the next president of IABS. Thank you, everyone. I now request Dr. Farzana Mehdi to present the vote of thanks.
बस काजिम भाई से क्या तो साइड हो गए थोड़ा सा और हल्का सा एकदम हल्का
आदाब, namaskar, and a very good evening to all the beautiful people amongst whom I feel privileged to be part of. Respected dignitaries on the dais, honorable guest of eminence, Padma Bhushan Professor Ashok Seth Sahab, who's Director Fortis Escorts, New Delhi, guest of honor, Padma Shri Professor Mansoor Hassan Sahab, Dr. Ammar Rizvi Sahab, ex-acting Chief Minister, Uttar Pradesh, Padma Shri Chunamani S. Gopal, all senior dignitaries of Indian Academy of Biomedical Sciences, Founder President Professor Hari Sharma Sahab, Patron IABS Professor Venkatesh Sir, President IABS Professor Raghu Sahab, Secretary General IABS Organizing Secretary Professor Abbas, Treasurer IABS Dr. Ranjana, Additional Professor Department of Biochemistry KGMU, and Professor T.S. Podial, who has joined us online all the way from Bhutan. Distinguished delegates, respected faculty members, and my dear friends and students. This is the last item in this event today, and I am standing in between you and your dinner, which has been served. Somebody has told me, and it is hot and piping and waiting for all of you. So I will not take much of your time, but I deem it as a great honor and proud privilege to propose this vote of thanks. Being the Vice Chancellor of ERA University, I take this privilege to thank each and every one, all the distinguished authorities who are sitting on the dais, and eminent guests from in and around Lucknow and out of Lucknow, and all the awardees and the recipient of the fellowship who have been honored today. I extend my gratitude that you all have come all the way in this hot weather of this May 2022, which is extremely hot by any means. I express my thanks to the organizers from KGMU and ERA who have worked together tirelessly and day and night during this month of April, and the entire program was put together within a very short span of 1 month. And this really is a phenomena, because this conference was being organized after a break of 2 years, and everybody had been waiting for this occasion to come and attend. But kudos to Professor Hari Sharma sir, who said that let us now organize this, and Professor Abbas took up the challenge, and his team from KGNU and my team from ERA, they worked in collaboration. So I would like to thank each and every one of you that it was because of all your efforts that we have seen this beautiful day today. And in the end, I would like to thank God for giving us this opportunity after Corona, that we all are sitting here and we all are attending this conference offline. So once again, I would say thank you very much. And for this journey, which we had started many years back, just one couplet. वैसे तो डॉक्टर मंसूर साहब बैठे हैं और प्रोफेसर साबरा बैठी हैं, इनके सामने तो शेर हम नहीं बोल सकते कुछ, बट फिर भी कोशिश कर रहे हैं क्योंकि लखनऊ से हैं कि ख्वाब तो देख अभी ख्वाब की ताबीर ना दे, ख्वाब तो देख अभी ख्वाब की ताबीर ना दे, और रक्स करना है, रक्स मीन्स डांस, रक्स करना है तो फिर पाँव की जंजीर ना दे, तो फिर पाँव की जंजीर ना दे. Thank you. All of you are invited to have your dinner. Thanks.
Before joining for dinner, requesting all to please stand for national anthem.
भूटान का बजाना पड़ेगा फिर. Yes ma'am. अरे ma'am, that's so generous.
जन गण मन अधिनायक भारत भाग्य विधाता, पंजाब सिंधु विंध्य हिमाचल तब शुभ नामे जा गाहे तब जय गा जन गण मंगल दा जय हे जय हे जय 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 Please join us for dinner.
Thank you, Era University and King George's Medical University. Professor Abbas, thank you so much indeed. And Professor Mehdi, thank you so much indeed.